GmbH, and with me today I have our key account manager Axel for our talk. He will be talking about functional bioassays for the development of therapeutic antibodies. I would like to remind that please feel free to use the chat function for your questions during our talk. I will be supporting that. And with this, I would like to hand over to Axel for his talk.
Okay, thank you, Asli, for the kind introduction. I'm happy that I can talk today about the functional bioassays Promega has developed. And the goal of my talk is probably to give you a very good overview about different methods and technologies which are out there from Promega, and not to bore you to death with too many graphs and something. So I hope it's constantly interesting for you. So first of all, I want to introduce Promega a bit, just one slide, because I don't know if everyone knows Promega. Promega was founded very early for a life science company in 1978 by William Linton, and it's still privately held. It's still our boss. And yeah, we now have over 1,600 employees in 60 different countries, but in all other countries, distributors. So wherever you work or something, you can provide Promega products, and most of the production, or nearly all production for the market here, is in the US, and we have some Asian production sites also, but they are covering the Asian market. And here are some pictures just to give you an idea where it looks like. And this is the Promega side, and nearly every product I talk about is developed here in this old R&D building. But I don't want to talk about Promega anymore. It's just maybe explain why we started to do bioassays. So Promega was dealing with luciferase assays from the beginning on, really cloning the first luciferases, and has really deep knowledge in luciferase reagent, creating reporter assays, and also had in hand all the reporters and very simple reagents. So it was low-hanging fruits at the beginning when the biologics came up. It's now 11 years ago when we decided to make the first bioassay with a reporter technology. So everything was there. We just had to put it together. And from the beginning on, our main goal when we made bioassays was lowering variability. Some of you might remember that if you only have primary assays in hand, that's a pain because you have very high variabilities usually. So lowering this variability to make it able to also be used in lot release and so on was our main goal. And there were several ways which led to this goal, but one I want to point out is the thaw and use cells. That's in principle cells which are used like a reagent. If you purchase a bioassay from Promega, that you can just put them out of the fridge, thaw them, and use them in the assay. That's it, no culture needed. And this lowers the variability drastically, but I will come back to this a bit later. And to bring us all on the same page, how an ideal bioassay looks like, this is things you know. Of course, it has to reflect the mode of action. It has to meet the ISH guidelines, or in terms of precision, accuracy, robustness, and reproducibility. Sorry. Of course, it has to be able for potency determination. Stability indicating is an important thing. And last but not least, it has to be good enough to, at the end, validate or set up a QC lot release assay. If you look at the physiological relevance of a bioassay, or assays in general, we have this graph shows pretty nicely that there is no correlation between quantity of assays you can pull through and physiological relevance. If you want to have very high quantitative data in terms of amount of samples, and so then biochemical assays fit usually best, and then the physiological relevance increases, of course, if you use cells, maybe 3D cultures or tissues, and at the end, of course, animal models, and in the very end, maybe then the human. All bioassays, which I will tell you now in the first part, are engineered cell lines. That is very good because they fit pretty well. They have a good alternative between quantity you can achieve and physiological relevance, because they really show the mode of action and you can, in principle, use them high throughput. Just to give you an idea where we can place the reporter assays here. I will also talk about the earlier and later stage in the end, but only quick. Yeah. In the meanwhile, I told you that Promega started 11 years ago with the first bioassays, and in the meanwhile, we have a great variety of assays for a lot of different targets. And this graph shows a bit the areas which we can cover. Maybe you find yourself here somewhere. Of course, FC effector activities are a big area of assays. Cytokine modulation, that's especially interesting for biosimilars also, like VEGF or TNF-alpha. Immune checkpoints are, of course, important more and more. Here we have a lot of assays. We can have tools for CAR T therapy development, and you can also use the assays, of course, for bispecifics or ADCs. To really guide you through and show you where we are, I have this picture here because it shows very well what we're doing. I will now talk about the genetic reporter bioassays. These are surrogate assays, which are excellent modified cell lines that can be used as bioassays. And there are two newer parts I just want to tease today. We have seen that there's beneath the reporter assays also a need of primary cell assays. Therefore, I would introduce you to our new area here, the primary cell-based assays, which have the same principle, that you can really easily just use them without cell culture. And even newer or as new as the immunoassays. Here are very interesting assays, for example, for FcRn binding, but also cytokines. That's a nice alternative to ELISAs, maybe. So I will start, and this is the biggest part, with the genetic reporter bioassays. You can divide the genetic reporter bioassays in these 4 fields. At least I did it. So FC effectors, immune checkpoints, cytokines, and T cell activation. So I will start with FC effector cells and take out one assay and explain it in more depth, because this is a good hint for the main principle Promega uses for bioassay development and bio. The first assay we've done now 11 or 12 years ago was an ADCC assay. This is one of the most spread assays in general, and there are several problems if you want to make a primary ADCC assays. First of all, you need NK cells. This can be cell lines nowadays, but also most people use PBMCs really from different vendors. And of course, if you take stuff from different vendors, you have a very high variability. In case of PBMCs, it's also the case that some vendors not even have ADCC activity. So you have a lot of variety, and maybe the assay does not work at all. Then usually you have to incubate your primary cells, NK cells, with your antibody and a target cell, which you also have to culture, and then you measure cell death. And there are several ways of measuring cell death. Typically can be LDH release, cytoxlo, GAPDH, or whatever, different methods. But however, you always have the problem that you have a co-culture and you measure a general death signal. That means the NK cell that dies also releases LDH. And as this is not in the body and nothing is transported away and you have it in the well, it might be that NK cells also die, especially if they are from primary source. So you never can be sure if you really only measure the death signal of the target cells. And these two parts give you together a very high variability. And at the beginning, when I talked to people, they were satisfied with about 30% variety between the assays or something. And this is not necessarily wrong. So the idea was to make a surrogate assay to replace this by a much better system. So what we have done, we took Jurkat cells, which are, of course, no NK cells, and modified them with everything they need for reflecting the mode of action of ADCC. Indeed, it's not so much you need to engineer here. First, we put in the Fc gamma R3A receptor, which is responsible for the binding, and then the subsequent ADCC. And we added a reporter gene and with an NFAT response element. This is how the way of ADCC, the signaling goes through. So, and then we have an effector cell that instead of releasing killing signals or kills someone, produces light. That's just in principle, if you want, one step before the usual killing would go on. If you now incubate this with an antibody and some target cell, you really just only get light. The target cells are not killed, of course, but you get only light. So it's a direct measure of the activation of the ADCC pathway. And this works very, very well. And one big part in lowering the variability, in addition to this variance of the viability, is that we provide this in a thaw and use format. That means you just have a vial where these cells are in. Principle, you can just plate it out and run the assay. And here's an example how easy it is in principle. You really incubate your antibody with the target cells, add engineered effector cells, incubate 6 hours or something, and then you add the detection reagent. So that's an ADCC assay you can really do in one day. And this is pretty not possible if you really only work with primary cells. And get very nice curves here. This is used by many different companies now. It's also used for lot release already. And what is also important, it correlates very well with real killing data. So this is a comparison of the reporter assay with the PBMC killing assay, and it really correlates well. Again, I want to point out the cells as reagents principle is key to all our bioassays. If you have a cell culture, you have to, of course, so the cells also culture them. You have to count, centrifuge, and so on. This all gives you variability. Even if you have 2 flasks and you incubate them for the same time, there might be little variations. What we do, we have big sizes of lots. We freeze them away with a very detailed method and have a very constant quality. So if you saw something, you can be sure that the variability is pretty low and you don't need any cell culture, plastic, media, time, or whatever. So that's really a big advantage. And in addition, you don't need to do all the analysis. So in principle, you have to do sometime SDR analysis to check the cell ID in your culture. You have to do mycoplasma tests, check sterility, and so on. This is all done before. And yeah, this assay was so well accepted that we said, okay, that's good. Then we don't change the principle. We do the same for all the other FCR receptors. So this is now a snapshot of things we have. Might not be 100% complete, but this is in principle what we have. We have here the ADCC part. We have the V and the F variant. The V is a high affinity variant, and the F is a low affinity, which is much more spread in the population. We have ADCP assays, the Fc gamma R2A, H and R variant, and Fc gamma R1, and also mouse ADCC variants. And this is a bit special case because the Fc gamma R2B is usually inhibitory. Here we change the the inner part of the receptor by an activatory one, and could do an activation assay by that. That's, if you're interested, we can talk about. So, but this is in principle the field of the Fc gamma R assays we made. So next thing in principle that came, because there's a large interest, is the immune checkpoint part. And in principle, we use the same principle. But of course, this is a bit more complex than only ADCC. So in this case, we sometimes need 2 engineered cell lines. So I will start with co-inhibitory because they're a bit more complex, and then look for the co-activatory targets. So this looks now very complex, but please don't be shy. I will go through it. So please forget everything which is on the slide and just look at the 2 cells here. We always, for these assays, have an artificial antigen presenting cell here and an effector cell here. And what is also common for nearly all of the assays is that we have in the antigen presenting cell this triangle here. This is a T cell receptor activator, which maximally activates the T cell receptor. And on the effector cell, we have the T cell receptor and a luciferase reporter with the response element of interest. So what in principle happens without all the other information on this slide is that these two cells are co-incubated and the T cell activator activates maximally the T cell receptor. So in principle, you get light. What you can put in now to create an assay is the different proteins here. For example, if you want to make an PD-L1, PD-1 assay, you clone in the antigen presenting cell and PD-L1 and PD-1 in the effector cell. And what happens now? In principle, you have a very activated cell, but PD-L1 binds to PD-1 here and inhibits the signal. So the signal goes down. So you have no signal anymore. If you want to check now for antibodies which bind either to PD-L1 or PD-1 here and interrupt this interaction, then of course you regain the signal and you get light again. And this principle works for all these targets in different combinations. And we even have cell lines which can measure two different things like PD-1 CTLA4, PD-1 TIGIT, and so on. This is the general base. So this of course works, but I don't want to go through I think Asli could provide the presentation afterwards. So if you want to see it, then you can have a look. I don't go through the graphs. I think that's rather boring to look for details here. And you can play around now. So for example, if you want to make a CTLA4 specific ADCC, you could take the ADCC effector cells and the CTLA4 effector cells, which have CTLA4, add your antibody, and then have a CTLA4 specific ADCC. And I go a little bit faster because the time is running. This is an example of a combination assay. If you want to look for combination therapies, for example. In this case, it's PD-L1 TIGIT. So again, you have this maximal activated cell and you have the PD-1 and TIGIT, which suppress the signal. If you now treat with, for example, here the blue line, an anti-PD-1 antibody, you see that the blue line goes up again. So you get some activity back. If you treat with anti-TIGIT, that's the green line. And if you take both, you really see a very nice synergistic effect of the both antibodies. Okay, and the next part is the co-stimulatory receptor bioassays. These are by nature a bit easier. And these are, again, just examples of targets we have. In principle, that's, again, one reporter cell line, which has the according co-stimulatory receptor, and, of course, ends, again, in a response element coupled to a luciferase gene. And what you can do now, you can activate them by a ligand, either soluble or bound or whatever, or treat with an antagonistic or agonistic antibody. And then you get a reporter signal that the cell is activated or not activated. Again, here are graphs that show the specificity. Of course, we have more data. One thing I want to mention here is that for the co-stimulatory reporters, it is sometimes important that we cross-link somehow the antibodies we use. We have some examples that the assay or general the bioassays in this field do not respond very well, even if the antibody is very good, because there's no cross-linking in your dish. So what we found out, you can go into literature also, there's from Sang and so different papers, and I haven't looked for one yet for especially this, so I'm sure there are more papers now. This cross-linking can be, for example, achieved by Fc gamma R2b. So we also have a cell line to co-incubate with this, just to have this Fc gamma R2b expression and have a cross-linking and then get a better signal. I don't have very nice example for real screenings. I have not included them here because of the time, but if you're interested in this, I can show you. Okay, then we go in a different order. The next is the T cell activation, and I hurry a bit. We also have cells for T cell activation. These are T cells which have reporters, and we, in principle, for the original version, we have two different reporters, one with an NFET reporter and one with an IL-2 promoter for things which are more CD28 related. So in principle, it's the same. You can these you can modify with proteins you want, or incubate it with a bispecific antibody, for example, and the cell with your interesting antigen, and then measure T cell activation. In the meanwhile, we also have different tools, like here TCR alpha beta knockout cell lines. We have CD4, CD8, and CD8 and 4, and other specific cell lines. This can be especially also interesting in CAR-T development if you want to put in your construct and look for activation or ranking or whatever. And last but not least, the cytokines and growth factors. That's especially also important for some biosimilars, but also in general. This is in principle, again, the principle of a reporter cell line. Again, we have the cytokine receptor expressed on the surface, either endogenously or engineered, depending on the cell line. This ends in a response element, and you get light if this receptor is activated. You can now stimulate with the cytokine you need, and then check antibodies which might catch the cytokine, or also if you have agonistic antibodies, which should also work. Again, all these assays, remember, are in this saw and use format, so you can just plate the bioassays, add your cytokine, incubate 5-6 hours, add the detection reagent, and read. So it's all one-day assays if you want, and very easy to standardize, because yeah, it's not many steps in principle. Yeah, and this is just an example of the activation by a cytokine, and then titration with an antibody. I have these things inside because in case you want to look at it afterwards. So this is now a never complete list of bioassays and targets we can cover. I think this is pretty nicely. That was from a colleague in the US who made that. It's pretty nicely with its 4 different fields. We have FC effector field here, and shows things. We talked about the reporter bioassays. I also want to mention this now in the next part here. Here are the immune checkpoint assays and T cell activation, and the bold things are in the catalog. The not bold but black things are in early access material. And this is, I want to explain a bit now. You will not necessarily see early access material in some publications or in the internet or something. So it is absolutely necessary that you contact us if you are interested in one of these things. Best via Ashley. You can also contact me, someone at Promega, and we can give you always an actual list of bioassays we have or working on. We have a special group which was nearly founded to the same time when we start with bioassays that should provide our technologies to our customers if they are not in the right form. And these are dealing now with all the development of the bioassays also. They are now called Elite Access. So most of the assays which are really ready and used by pharma companies and so are not in the homepage or catalog yet because of some marketing stuff or something, but they are ready to use. You can order them, you can use them, but the list is not necessarily on the homepage. So please ask us if you need a special target. And this group is now specialized since 12 years only in developing reporter assays and also screenings and other things. So even if we don't have the target, we usually have a lot of projects where we just create an assay for someone, maybe make first a feasibility study and then go on. So this might sound. So please contact us in any time you are interested in something like this. So as promised, time is running. I want to drop some words with a new field we have, the primary cell-based assay. So why do we do this? In the first hand, we say reporters are the better bioassays. Yes, in principle, they are less variable, but at the end, the FDA or other authorities wants to see maybe real killing data for ADCC, or at least bridging experiments that show that the reporter assays show the reality. So we are back and have to do a primary assay. So what can we do to make it easier to make an ADCC assay with very low variability and that works 100%? For that, I just have to introduce 2 basic technologies. First of all, that's not new to us that we think about how we can measure in a co-culture that one cell is dying. So we want to create an assay which can distinguish between two cells you're killing. I just go ahead. If we now look at the ADCC in principle, our idea is that we take pre-qualified thaw-induced PBMCs. So it's really from vendors. We pre-qualify them that they make ADCC and that they are in a good range, and then we freeze them away so you can use them again as a reagent. Just take the ready PBMCs and plate them out, and that's a big advantage, I think. And combine it with a target cell killing assay. That means we take a target cell which is modified a bit to makes us able to measure only the cell death of the target cell. And how does this work? Therefore, I have introduced something else. So all I tell you now is just to understand the technologies which come now. You're probably all familiar with the firefly luciferase, but it's not the only luciferase around. And some years ago, we discovered a new luciferase, and this is from Oplophorus. That's a deep sea shrimp that spits out clouds of light to its predators and therefore blinds it, just like an octopus in higher regions. And it's also a bit sticky, so it marks the predator for being eaten by even bigger predators. This was originally a tetramere, and there's a long story. I don't tell them. We just make directed evolution and substrate evolution. So at the end, we ended up at a monomer with 2.5 million brighter than the original. And it turns out that this luciferase is very small. It's very blue. It's very fast, and it's very bright. So it's very, very protein compared to the others. And this gives us completely new features to build assays. That's in principle, it's a good reporter. So, and then it turned out, or we find that if you take out a fragment of only 11 amino acids, here it's 13, but in principle it's 11 amino acids out of this, then the luciferase is not active anymore. And you can put it back and have a classical complementation assay where you gain back luciferase activity if you put these two fragments together. What our scientists have done now, they took out this part and again modified it in different ways. And what came out is that we find that we have a lot of different fragments here that work, but that have very different affinity to the large fragment, different Km values. What we did now, we took out the fragment we found with the highest affinity and with the lowest affinity, because these are great tools to build assays. The one with the low affinity, that's the small bit here, gives us the possibility to have a great tool for tagging proteins to measure protein-protein interactions, because it nearly has no effect. It's very small. And the high bit, the one with the highest affinity, it really binds and makes us possible to quantify things. And the assay I will tell you now is based on this high bit. So we have a very small tag with 11 amino acids that binds very hard to the larger part, and then you get light. And the detection is also very easy. You can just, you have the high bit expressed somewhere, the very small fragment, and then in the detection reagent, you usually have the large bit, the bigger part can be complemented. The substrate is also inside, and then you get a very nice light, and the light is nearly as bright as the original nanolook then. It's very sensitive. I don't go into depth. So how that killing assay works now. So in principle, I have a co-culture of an effector cell and a target cell. And the target cell is now modified with a halo tag protein. That's, again, a new technology I won't explain here. It's just an absolutely non-human protein which has no effect in the cells, which carries this 11 amino acid part. And what happens now is in principle a classic killing assay. We have expressed this constantly in the cells. And if now the target cell kills, this can be released, this halotech high bit fragments, and then you can quantify them by adding the individual reagent and have a very nice quantification of exactly the cells you want to measure. And this is now how the combination looks like. In principle, you could plate your hybrid target cells at the PBMC cells. If you use the frozen ready-to-use pre-qualified from us, you again just have to open a box, put it in, add the antibody, wait 6 hours, and have a wonderful primary ADCC assay. That's fantastic. And yeah, this is again, the time's running. So this is again examples how well this correlates with different antibodies, trastuzumab and cetuximab, how good this correlates between the reporter and the primary killing assay. So this is an overview about all these primary things I can't mention today. Or you see here the functionality here. We have PBMC ADCC assay. We also have TDCC assays with primary cells, ADCP, and here other different target cell lines. We have many more. So these are target cell lines which are already modified with this hybrid, and also, yeah, the effector cells can be purchased. So that's just for an overview. Good. Now I have about 10, 15 minutes left. And this I want to spend to show you a new technology, which is, thank goodness, based a little bit on the same technology, so I don't have to explain it again. Called LUMIT or LUMIT immunoassays. And this here we use the wonderful features of this small bit I showed you. That you mean the large fragment with the one with a low affinity to measure protein-protein interactions for immunoassays. There are three different principles. You know now how it works. The idea is to tag antibodies with it, like for Eliza or something. So the first method you could imagine is that you just take your primary antibodies which bind to an analyte. Of course, they have to bind to different binding sites. And then if they both bind, they combine and you get a very nice light in the presence of the analyte. The second idea would be to use secondary antibodies. So we have pre-labeled secondary antibodies. So if you have an analyte and you have good antibodies for it, let's say you have a kinase and a phosphor antibody, and you want to look if the phosphorylation is there and both bind, you can simply add secondary labeled antibodies. That means whatever anti-mouse, anti-donkey, or anti-cat or whatever antibody, and then you get light if these two antibodies build. So you can build your own assay pretty easily. So this is, again, not all predefined. We have kits where you just can label your own primary antibody and establish an assay. That's maybe the cheapest but more work version. The secondary is and one very nice thing I want to show you is the competitive binding. In this idea, we have an analyte, which we call a tracer, something. This is something where you want to measure that something binds to. And we have an antibody against this tracer with a large bit. So in principle, you have light in your reagent, and then you can titrate with something that competes with this antibody, and the light drops down. And one very nice example is our FcRn assay here. This is now from the physiological relevance a bit deeper than the cell lines because it's in principle biochemical assay. FcRn is responsible, FcRn binding is responsible for how long antibodies stay in the body because it's in the endosomes. If they survive the endosomes and are pre-released, if they have higher binding capacity. And the assay works like this. The assay reagent, you have this human FcRn protein in solution with the small bit tag. This just is 11 amino acids which bind with low affinity to the large one. And you have an antibody with a medium or low binding to, or medium binding to the FcRn, which holds the large bit. So if you put this together, you have light because the luciferase is active because the antibody binds here. Now you can take your analyte antibody and titrate it via this, and you will see the signal dropping the better the binding affinity of your analyte is. And this gives you really a nice assay, plate-based assay, where you can measure or compare FcRn binding affinities. These are, again, data for this. It works. And yeah, I go through this a bit faster because I already explained the technology. The principle is for the LUMIT bioassays, where we want to check for cytokines and so on, we use the primary principle here that we have two antibodies to an analyte which are labeled with large bit and small bit, and then get light. And this you can use, for example, whatever, if you want to use this for TGF-beta or whatever detection. You can use it in principle like a plate-based ELISA. If you compare it to traditional ELISA, and this is a very nice slide which a colleague from me was doing. For a normal ELISA, you need a coating, washing, blocking, washing, analyte addition, washing, addition of the first detection antibody, washing, additional second detection antibody, washing, substrate addition. Takes forever. I think nobody likes to do ELISAs, even if you like the results which come from them. Doing ELISAs is, in my opinion, a pain. So imagine that you can, just by adding these two antibodies, just get rid of the coating and blocking steps. You can also remove the washing steps because you just have to add the antibodies. And you don't need two detection enzymes or something because the luciferase is included. So this limits this all ELISA process to only 60 to 90 minutes to have a really high sensitive, great immunoassay, which has really comparable data to the ELISA. And this is a very fast growing field here. Slides can never be actual, so that's the principle again. And first of all, we have a toolbox where you can label your own antibodies, though, if you don't have something. And there are a lot of ready-to-use kits. This is not nearly complete. We have much more than these targets in the meanwhile. I mean, it's also TNF-alpha and so on. So that's very fast growing where we have ready-to-use assays you can just use. And yeah, please feel free to contact Asli. Maybe we can arrange something that you compare it to your ELISA. So I'm very comfortable that you would be happy with the easiness of this and the time saving. And we also have glucagon insulin that might not be too interesting for you. So in principle, I am a bit faster than I thought. That's good, but I'm not to the end. Just to round this up, Promega has, in addition to this really classical bioassays or immunoassays, a great variety of tools for mass spec sample preparation. I mean, maybe you know us as a supplier for trypsin, but we also have a lot of different proteases. And I want to point out some of these products because they are very interesting, especially maybe in the biosimilar or biologics development. Of course, trypsins and so on are known by you. We have this wonderful product which we can digest at very low pH and thereby inhibit post-translational modifications which come from the digestion or from the preparation of a protein. If you want to look at an antibody and make benchmarking stuff and so, or looking for aging, you really want to see exactly what is on the antibody and not some modifications you accidentally put in by different detections. And this is really absolutely made for this, that you have no post-translational modifications by your digestion. That's great for really for benchmarking and so on. Look at it. I can also say we have this IDES and IDES, which are for Fab fragment isolation. And another thing I really want to point out is a completely new protease for people who make mass spec here. We have a specific proalanase, and this is, to my knowledge, still new and the only protease which binds cuts specific at this pieces. So this can really help for one or the other question in your field. And of course, all the other proteins we have around. Also, we have reference mixtures. So please go ahead if you want to have information. Contact Asli. I will show you the contact information soon. So this slide you know already, and I really want to summarize what I think is the most important take-home message you could have. So first of all, I think it's nice to understand and see that we have different fields of bioassays we can provide. The first is the genetic reporter bioassays, which was the biggest part at the beginning. And here we really can cover the Fc gamma or the Fc gamma R assays, immune checkpoint, and so on. And we are open to develop for you, or sometimes we have something in the fridge already which we can provide, or even if it's just information that you plan to do in bioassay, like, and maybe we already tried it and it didn't work, and this can be a very helpful information also. Not always, yes, we have, but sometimes we tried and it didn't work is even more important, can be more important than, yes, we have. And it's what is here the most important thing is that we have this in this saw and use format. Again, to explain, you really just have a vial inside which you do not propagate. You just put it into your assay and use it. Therefore, have nearly no variability. Then the second part is that not Promega only makes reporters. We start to do go in this primary assay field and try to help here also to lower variability. It makes it easier by also providing thaw and use cells. Again, this just thawing and taking as a reaction to help or to help with samples. And the third part, the immunoassays, one I think which is absolutely direct related to the biological is the FcRn activity, but also all the other cytokines you could measure if you want to check or compare with your Eliza, please contact us again. So, and last but not least, this is Asli. I don't know if you see her in the chat also. She's very experienced and very kind and would be your first contact. Of course, you can contact me also, but in the end, Asli is the main contact. And please don't hesitate to ask her. We always can help. We not always can. We always try to help. We can't always say. Good. Okay, then I'm at the end, 3 minutes earlier. So please, I don't know how we do it with the questions in the chat. The chat is full of questions. Please feel free to use the chat question if you want to ask questions. I will be still here now for some minutes. I think we are thrown out at 10:50, so so long. We can wait. You can also say, was this interesting at all? Yeah, I can already say thank you very much for your attention, at least. I don't see how many people joined or not familiar with this tool. You're welcome. Yeah, Dilara, if you that's great feedback. Thank you. So if you want to have more information about that, we can even, if you don't want to talk, we can even send you something or information material. That's really great. People who use it love it. Usually, it's a pretty new product. This will be more present in the future. But yeah, it's really good to have only 90 minutes in case of 2 days. Yeah. It's a lot. I have the full list of availability. It's really changed every day. They are working on it like hell. But I think we have different species, for sure, not all, but for different species. Invertebrate, yeah, that depends on how good the binding is. We really should have in detail, because it depends a bit on the antibodies and the similarity of your targets to human ones. I would say in invertebrates, that's usually not very similar. But you could use it anyway, because you could label your own antibodies very easy. And it's a little bit more work, but you could set up really easy. If you have 2 antibodies against one target, you could just label the one with large bit and small bit. Several people have done it and no one failed so far, so it's really good. Yeah, Leil, we can update you or send the presentation also afterwards. I also give private presentations if wanted.
Yeah, if you can contact me through my email, I would help you for the presentation. And I also would like to thank Axel and Mihaela very much for supporting this.
Yeah. Yeah, maybe you can contact Dilara or otherwise to give her
Sure.
more information about the Lumit, because that's also very interesting, especially if you, the good thing is if you don't have targets which for human or something, especially invertebrates, and you have good antibodies, it's hard to get an ELISA anyway, right? So that could be a very good idea to just use this pretty easy, and I think it's not very expensive, even this labeling kit to create your own assay. Or you use secondary antibodies. Try how it works. If you have whatever, an anti-goat and anti-mouse antibody, and your assay is standing, you can just try to use the secondary ones. Sometimes it's not a big difference. Sometimes the secondary ones have a little bit lower signal, but only sometimes. That's yeah, I guess we are thrown out in some minutes. 2 minutes.
If you want, you can actually leave the session via the orange exit button on the right hand side.
Okay, just yeah, I think maybe we can leave that. Not, I don't see many people still, right? So there shouldn't be questions. So we leave. Thank you very much, everyone, also for the possibility, and have a nice day.
Yeah, thank you very much for
I leave
attending.
now. Bye.
Thank you. Bye.
Currently, we are live. I will be leaving the floor to
Okay, shall I start? Good afternoon to you all. I'm Berna Özkan and I'm working as marketing manager at Antechnik. We will be hosting a series of seminars during BioExpo Online. First of all, I want to thank to the organizing committee for their efforts in transferring BioExpo into an online platform once again during the pandemic times. And I would like to thank you all for attending this session. We will co-host this session with Thermo Fisher Scientific. Antechnik is the authorized distributor for Thermo Fisher bioprocess product line. Today's title is High Performa Dynadrive Disposable Bioreactor Transformational Technology, and our speaker will be Floris Hojenga. Floris has completed the Bachelor of Science degree in Clinical Chemistry, and after that he worked for the biopharmaceutical industry for more than 10 years. Then he started his commercial career first at Inforce Benelux and Pines Solutions, which got acquired by Thermo Fisher Scientific in 2017. He's now, in his current role, he's the commercial sales leader for bioprocessing equipment and automation portfolio in Americas and EMEA, and he's supporting the single-use technologies team. Floris, welcome again. May you start your presentation, please? Thank you.
Thank you, Berna, for the nice introduction. Yeah, good afternoon, everybody. My name is Floris Hojenga, and I'm the commercial sales lead for Thermo Fisher's Scientific bioprocessing equipment and automation portfolio. The next 30 minutes or so, I would like to tell you more about our latest innovation, the Dynadrive, and highlight the features and benefits of this transformational technology. Before I dive into the Dynadrive, I would like to give you briefly an overview of Thermo Fisher Scientific as the world leader in serving science. We have more than 75,000 employees globally in all areas of science, of which 10,000 customer-facing, so we can always be there for our customers. With 5,000 scientists or engineers, we have focused experts and scientists in-house that are focused on the same things, no matter what the science. We are investing $1 billion in R&D as we are committed to advanced science and staying ahead of innovation. The Dynadrive, which I will tell more about, is a good example of this innovation. With more than $25 billion in revenues, we are not going anywhere, which gives you the insurance policy that we stay around to support you. At our core, we are scientists, and our purpose is to enable our customers to fulfill their scientific purpose so they can make the world healthier, cleaner, and safer. Looking at the market dynamics, there are changes occurring in the rapid growing pharmaceutical market, evolving landscape. An example of this is the rise of generics and biosimilars, but also breakthrough science like CAR-T. Shift in economics. Pricing was always important, but it is getting even more important. The Dynadrive can be really beneficial in reducing cost, on which I will elaborate further in this presentation. Digital disruption, like automated workflows to drive process speed and efficiency. This is something Thermo Fisher has great experience with within our bioprocessing equipment and automation portfolio. Thermo Fisher is offering our customer a comprehensive in-house or outsourcing solutions from discovery and research to process development and optimization, to clinical trials manufacturing, clinical trial supply and logistics, and finally commercial launch, all based on a foundation of quality. Within the bioproduction offering of Thermo Fisher, we have products integrated into hundreds of clinical and commercial pipelines. Some of these products are our Gibco cell culture products, our chromatography and purification solutions, quality control testing, production chemicals, and our single-use technologies. With our offering, we support our customers from early phase development through commercialization. As we have decades of experience, we help improving the productivity of bioprocessing and creating solutions to overcome challenges. With doing this, we enable process intensification in the bioprocessing workflow without compromising on quality. We want to drive performance by collaborating to achieve the optimal bioprocess outcome for our customers. Finally, before we dive deeper into the high-performer Dynadrive, I want to show you the overview of our best-in-class single-use solutions offering. Our single-use technologies consist of three parts. Flexible containment, in which we offer standard and custom bioprocess containers, or BPC like we call them, based on our well-known Aegis 514 film. For critical sterile liquid handling in 2D and 3D format. This also includes fluid transfer solutions, also known as line sets. Secondly, rigid containment. This includes carboys, bottles, critical environment products, and adherent cell culture solutions, with in mind to reduce process time and enhance flexibility. Thirdly, our bioprocessing equipment and automation portfolio. This includes innovation and customized solutions for automation, software, and controls, bench-scale bioreactor systems, single-use bioreactors, single-use fermenters, and single-use mixers, and transport solutions. The Dynadrive falls also in this portfolio. Our single-use technologies can be used across the workflow, from upstream to downstream processing. Let's dive deeper into the high-performer Dynadrive, because that's why you're all here. Some of you may be wondering why Thermo Fisher Scientific made a next-generation single-use bioreactor. After all, Thermo Fisher Scientific has been providing single-use bioreactors since 2006 and have over 1,400 installed globally. Well, we were seeing some challenges occurring in our industry, like the need to support intensified cell cultures, like in fed beds and perfusion, which require improved mixing and mass transfer performance. Also, there's a need for flexible multi-product facilities that allow for the bioreactors to be flexible, scalable, and efficient. And finally, there is a need for larger single-use bioreactors, larger than the 2,000 liter we see nowadays. As many processes go from clinical to commercial phase, we found that our users would like to stay within a single use, but often cannot due to capacity or performance constraints. Therefore, we sought to create a new bioreactor that could meet all of these needs. We created the Dynadrive. This project has been in the works for nearly 4 years, and we are excited to report that all sizes are launched and now available. The Dynadrive reactor has 5 key benefits that I will highlight throughout my presentation. The first benefit is best-in-class performance in power mixing and mass transfer. The industry is looking for a reactor that has the ability to support high cell densities and that is accomplished with high yet efficient power and mass transfer. We are able to have similar performance at all scales. The second benefit is efficiency facility footprint. As you can see in the CAD images on the right, the Dynadrive is not only the 50 liter, but we offer a 500 liter, a 3000 liter, and a 5000 liter size as well. All sizes fit in a relatively small footprint. You will see in some of the subsequent slides that I present that the 50 liter is a great scale down model for all sizes. Also, that the 3000 liter can fit in the same footprint and ceiling height as our current high-performer sub 2000 liter reactor. Therefore, it can be used in most facilities that are built for 2000 liter reactors today. The third benefit is an impeccable C-train turndown ratio that allows for more streamlined and efficient C-train processes. I cover that in an upcoming slide. Fourth, we designed all sizes to have a lift mechanism to help facilitate bioprocess container loading, or BPC. The 50 liter and 500 liter have a bit of a simplified lift system, but the 3000 liter and 5000 liter both have a lift system that allow the BPC to fully connect it and load it at ground level, thus reducing the time users spend at the top of the reactor and also facilitating faster load and unload times. Fifth, we designed the hardware with the future in mind. There's quite a bit of space for connectors for sensors and connectors that are used today, but other may come in the future as well. So we kept that in mind when designing this bioreactor. I won't spend a lot of time on this slide, but just know that a lot of thought around ergonomics went into the design to make sure that it was easy to load and unload the bioprocessing containers. Like all our bioreactors, also the Dynadrive can be controlled by our high-performance G3 Lite or G3 Pro controllers. This will facilitate that all systems can be controlled by our TrueBio software running on Emerson Delta V. Similar features were designed in a 500 liter, 3000 liter, and 5000 liter as well. We do have a standard BPC which utilizes the Thermo Fisher Scientific Aegis 514 film. These standard designs have now been used by approximately 20 customers in various processes, and we have found that the standard fits almost all applications. We are using single-use sensors and foam probe in these standard designs. This standard design is similar to the one I just showed you, but it includes a 1-inch port in the middle to facilitate the connection to an ADF filter or TFF filter. Evaluation users used a design very similar to this one and sometimes only needed to some minor changes on the line sets. We will continue to customize BPCs as needed, but are also confident that our standard BPC design will meet almost all application needs. I'm showing the 50 liter standard BPC, but we have similar designs for the 500 liter, 3000 liter, and 5000 liter as well. We've talked enough about the 50 liter Dynadrive. Let's move on to the larger sizes. This slide shows you in relative dimensions the differences between the existing 2000 liter reactor and the new 3000 liter and 5000 liter Dynadrive reactor. As you can see on the left side of the slide, there are only 3 sizes which can accommodate the full seed frame. I will talk more about this in the next slide. With this, you can reduce the amount of floor space needed, but will increase your capacity. Also, I want to point out how the 3000 liter Dynadrive, shown in the right in the light blue, has a slightly lower required ceiling height than the ceiling height required for the existing 2000 liter reactor, which is around 3.7 meters. This means that a 3000 liter Dynadrive will be able to fit in the same spot where an existing 2000 liter sits, therefore boosting capacity by 50% and not taking up more space in the production area. You can see that the 5000 liter is a bit taller, roughly around 4.7 meters, and may be more suitable for greenfield opportunities as this requires higher ceiling heights. Now let's take a deeper dive into the Dynadrive technology. Inside the BPC is a multi-impeller drivetrain. The drivetrain is flexible when not deployed in the reactor, allowing packing to be quite small and compact. However, when the BPC is loaded in the hardware, the drivetrain becomes rigid as it's held in the top by the motor and at the bottom is held in place by utilizing a blind bearing port, while the corners of the BPC are held by the tab holders. Through many studies, including the empirical data and CFD modeling, it was determined that we can achieve the best mixing performance for the height and shape of the reactor by having multiple two-bladed impellers and a bottom sweep impeller. The sweep impeller allows for the incredible turndown ratio that I spoke about previously. The technology enables 10:1 turndown ratio for the 50 liter Dynadrive, which means you can seat the reactor with as little as 5 liter. The 500 liter and 5000 liter units can be seated with 25 and 250 liter respectively, yielding a 20:1 turndown ratio. This design also allows the Dynadrive to have a flexible terminal or end stage volumes with the same vessel. We are gathering the KLA data at various volumes within these reactors. Oh, yeah, sorry for that. Here are some more images showing how the BPC is loaded in the 500 liter hardware. Also on the far right, you see a CAD drawing of the lift mechanism used on the 3000 liter and 5000 liter reactors. This lift allows for BPC loading at ground level. Users will connect the BPC to the motor and also connect the corners to the top back and top front corners. The exhaust filters and condenser are also on this lift mechanism. They will also connect a foam probe, pressure sensors, overlay gas, anti-foam feed tube through this lift. All done make installation as easy as possible at ground level, even for the 3000 liter and 5000 liter Dynadrive. As discussed earlier, CFD and empirical data was used to determine the location, size, and shape of the impellers. We found that comparative mixing times improved when using 3 or more impellers when compared to using 1 impeller. This combination of multiple impellers, optimized design, and location resulted in a distributed efficient power input, low shear, optimized mixing performance, optimized sparse gas dispersion, minimized mixing times, and improved sparse gas-free entrainment, all to facilitate higher cell densities. We get a lot of questions about the shape of the Dynadrive. The shape is square, or like we call it, cuboid. This is done on purpose because we found that we can get superior mixer and thus more efficient performance by utilizing the shape. As you can see on this slide, each vessel shape does have some advantages. Moving from the left to the right, the worst case is a centered cylindrical. This design can yield unidirectional flow, vertexing, and poor mixing times. The off-centered angled is what the legacy Thermo Fisher Scientific sub utilizes. This design is good, but can cause some unidirectional flow, and we recognize that this design is satisfactory for the 2000 liter scale, but would not have been possible to scale up to 5000 liter. Probably the best design is cylindrical with baffles, like what you see in stainless steel vessels. However, this design is difficult to do in single use, especially in large single use vessels. We would have to put in baffles in the BPC or on the vessel and expect the BPC to conform around the baffles without wrinkles and folds. That leads us to the cuboid design, where the corners act as baffles and disrupt the flow, therefore providing disrupted flow patterns and no vortex formation. The impellers are also off-centered, which will help the bulk turnover within the reactor. Also, this leads to better mixing times and superior mass transfer performance. Speaking of mass transfer, we are utilizing the drilled hole sparge, or DHS as we call it, in all sizes of the Dynadrive. The DHS is not new. We introduced it in our subs in 2014. However, it has been enhanced with smaller, but more holes, thus producing smaller bubbles which improve the oxygen transfer rate. But also the bubbles are of proper size to also strip the carbon dioxide. It has been proven that the DHS provides more than adequate oxygen transfer, but also strips the CO2 effectively as well. This performance is seen at all scales. The other benefit was mentioned is that the DHS has a linear performance, as shown in the graph, in the dark blue line. Fridge parts provide a lot of oxygen mass transfer with low flow rates, but then they plateau and are not linear. The DHS, however, give much more better scalability across the range of the Dynadrive sizes. We definitely prefer the DHS, and we know you will as well. I've mentioned the C-train turndown ratio a few times in this presentation. This slide shows the benefit of the turndown quite well. The top graph shows a traditional C-train from vial to 2000 liter reactor, with multiple steps in rockers and smaller reactors. Now, if we compare that to the C-train of the Dynadrive from vial to flask, 50 liter and then 5000 liter, this process has been done on a numerous occasions with our test bioreactors and has been proved to be very effective. You can see that this improved C-train reduces the number of connection points, which reduces the risk of contamination or failures occurring. It also reduces labor and the number of consumables that are needed. This also reduces a lot of waste by more than 2 times. Not only does it reduce the number of BPCs, but it also reduces the shipping, warehouse, and packaging of these BPCs. And I want to speak about examples that we have seen while using the Dynadrive. Recently, one of our customers shared in a webinar the results and how they were able to exceed 200 million cells per milliliter in their perfusion process. We have also seen cultures exceeding 300 million cells per milliliter. We have demonstrating seeding a 50-liter Dynadrive directly from a high-density cell bank file. Also, there is a co-publication that we have done with Regeneron. It shows the CFD analysis comparing the 5000-liter Dynadrive to the 2000-liter legacy high-performance sub, and also 10,000-liter stainless steel vessels. It's worth a read. We will continue to collect and share more data. Some data that will be coming soon is KLA data at all sizes and different volumes, and sparser rates within each size of the Dynadrive. The data shown in this graph on the left shows well the KLA data for the 5000 liters shown in the light blue and the 50-liter Dynadrive shown in the yellow, aligned with each other. This performance also shows how we can be confident in processes that are evaluated at 50-liter scale will scale quite well up to 5000 liter as well. This data set also shows how the Dynadrive KLA compares to the legacy 2000 liter sub, shown in the dark blue line. The Dynadrive does have improved KLA performance, but also can be tuned by adjusting the sparge flow rate to also match legacy process requirements as well. Other data that will be published in an application note soon is some cell culture data of different show cell lines that have been cultured in the 50 liter, 500 liter, and 5000 liter scales. On this slide, you see the data of the show as fed batch process, maximum cell density, the viability, and the titers match very well within all three sizes of the reactors and were also very comparable to when cultured in legacy reactors as well. Hopefully this presentation so far has given you a better understanding about the Dynadrive design and more about the key benefits that the Dynadrive offers. It's definitely more than a reactor that enables high cell density cultures, but it can be used effectively for fed batch processes and enables some unique and valuable advantages when compared to other reactors. To summarize, best-in-class performance in power mixing and mass transfer, efficient facility footprint, high turndown ratio that allows for a more streamlined and efficient seed train process, ease of use and BPC loading. The hardware is designed with the future in mind to increase flexibility. So far, I talked quite a bit about the technical benefits of the Dynadrive single-use bioreactor. I now want to briefly highlight some of the financial benefits of using these larger scale reactors. There was a special report published in Bioprocessing International in October 2020, and we also had a webinar introducing the financial benefits of using the 3000 liter and 5000 liter Dynadrive vessels in November 2020. I just want to highlight a summary of our analysis. Through our calculations, we found that users could see up at 25% lower cost per gram when operating a series of 5000 liters Dynadrives, when compared to equivalent throughput from a 2000 liter sub. These cost savings are driven by the reduced number of batches and reduced cycle times and increased throughput. This file can be found on the BPI side. This chart shows planned output kilograms per year versus number of reactors to achieve that output. The different colors represent the output based on different reactor size and quantity of those reactors. For example, the blue line is a typical 2000 liter bioreactor, so capacity is added at 2000 liter at a time. Dynadrive is the orange and gray line for the 3K and 5K respectively. The yellow and the green lines are stainless steel reactors, 10K and 15K. You see on the x-axis the amount of reactors and on the y-axis the max kilograms per year. We chose to focus on comparing the throughput when operating 6 2000 liter reactors, which is quite commonly used nowadays and which some customers call 6-packs. When operating these 6 reactors with a 3 gram per liter process, a facility should be able to produce nearly 400 kilograms per year, as shown here in the blue dot. In order to produce the equivalent amount of material, users could operate 4 3000 liter reactors, as shown in the orange dot, 4 reactors, or they could operate 2 or 3 5000 liter reactors, as shown here in the gray dots, to generate roughly the same amount of 400 kilograms per year. Alternatively, users could also operate one 10,000 liter stainless steel clean in place and steam in place vessel that is shown here in the yellow dot. So this basically compares all the systems with around 400 kilograms per year. Let's keep that 400 kilograms per year in mind when moving to the next slide. So if we keep the 400 kilograms per year, like I just explained on the previous slide in mind, we equilibrated on that amount in these coming two graphs shown here on the picture. In the left graph, you can see on this slide a 27% reduction in OPEX per kilogram per year when operating 3 5000 liters compared to the 6 2000 liter subs. These are the 6 2,000 liter subs, and these are the 2 or 3 5,000 liter subs, which will result in a reduction of 27%. In the graph on the right, you can see that when you add in capital cost, the cost per gram is reduced by roughly 25%. So if you compare the 6-pack of 2,000 liters and compare it to 3 5,000 liter, you see a reduction of 25%. Or a 41% reduction in cost per gram when compared to a 10,000 liter stainless steel option, including 1% weight average cost of capital over 8 years. Drops from here to there. This concludes my presentation. Hopefully you can have a better understanding about the Dynadrive after this presentation, including its features, benefits, and value that it can provide. I will now answer some questions from the chat. Please drop your questions in the chat if you have any. It looks like that my presentation was quite clear. I'm happy about that. There are no questions. So, okay. Sorry.
Thank you, Floris, for this nice presentation. Thank you.
Okay. Thank you all for attending, and I wish you a great day.
Okay. Thanks.
Bye.
Bye-bye. Maybe we can wait. We're live now, Tom, but maybe we can wait 1 or 2 minutes for it to come. Okay. Okay. I can start now.
Yes.
Okay. Good afternoon to you all. I'm Berna Özkan and I'm working as marketing manager at Antibodies. We will be hosting a series of seminars during BioExpo Online. First of all, I would like to thank the organizing committee for their efforts in transferring BioExpo into an online platform once again during the pandemic time. And I would like to thank you all, of course, for attending this session. This is our second session today, and we will co-host this session with Optical Biotechnology. Antibodies has been the authorized distributor of Applicant since 2015. Today's title is advances in single-use upstream bioprocessing, new products and applications. And our speaker will be Tom van Aragon. Tom is working at Optical Biotechnology as a product manager for the single-use bioprocessing equipment. His role starts from benchtop bioreactor scale. His background is in mechanical engineering, R&D, and process modeling, and he has a master's degree in mechanical engineering from Delft University of Technology. Tom has been working for Applicant for over 4 years. Tom, welcome again, and thanks for your time today. May you start your presentation, please? Thanks.
Thank you, Berna, for the kind introduction. Glad to be here again. I had the privilege of presenting in last summer's session, also a section about Optical Biotechnology and our product portfolio. And today I want to go into a bit more detail on the advances that we've made in our single-use portfolio. And we will do that by looking at some of our new products. And we also will give the floor to a scientist that have been working with our bioreactor systems for a cell therapy application. So with that, I'd like to start the presentation. If there are any questions, we have the chat window and we'll be able to talk. So without further ado, let's get started. We're going to start off with giving Optical Biotechnology a short introduction first. As you can see here, it says part of Getinge, and that is because Optical Biotechnology has been acquired by Getinge beginning of 2020. We will now go through this introduction, and if there are any questions or remarks, please remind that you can put them in the chat window and we'll be able to discuss those shortly. Optical Biotechnology is part of the life science group within Getinge. This life science group is there to serve our life science customers. For these, we offered washers, sterilizers, isolators, and with the addition to Applicant to the group, now also advanced bioreactor systems. If we look at Optical Biotechnology within the Getinge group, we have the same overview as we just seen. Optical Biotechnology has been founded in 1974. We purely focus on upstream bioreactor systems, from research to production. We have done that since the day we were started, and we are doing that until now. About 80% of our business goes into pharmaceutical industry and 20% goes into industrial biotechnology. We're ISO 9001 certified, and our headquarters is located in Delft. Combining all the people that work in Optical Biotechnology, we have over 180 people working with Optical Biotechnology worldwide. We have our own branches in the US and in the UK, and the rest of the markets we work in with distributors. We supply advanced bioreactor systems to the life science market. These are bioreactors, but also the bioprocess control towers, which are next to the bioreactors. Also, we offer bioprocess analytics, meaning sensors, and software to be able to turn your data into information. Additionally to this, we also have perfusion systems available. As we just seen, Optical Biotechnology provides into pharmaceutical industry and into industrial microbiology. In the pharma industry, we supply to vaccine development companies, drug development companies, but also novel companies, startups, working in the field of regenerative medicines and cell and gene therapy. In the industrial microbiology, we see that we supply equipment to companies working for biofuels or biochemicals, or universities doing research towards recycling. We have just discussed Optical Biotechnology within the Getinge group. As mentioned earlier, if there are any questions, comments, or remarks, please leave them in the chat and let us know, and we'll be able to answer those now. If there are not any questions, we will continue with the Applicant product line overview. Here we will have a look at the products that Optical Biotechnology has to offer. If there are no further questions or comments, we'll continue. Optical Biotechnology is a full solution provider. So what do we mean with full solution provider? That means that we can supply equipment all the way from the smallest scale in lab research, all the way up to large scale cGMP production. We start here on the smallest scale. Our smallest fully controlled bioreactor is 5 milliliters in size. This comes in the form of the micromatrix, where we have 24 bioreactors in a cassette, which are operated under fully controlled conditions. If we take one step bigger, we go to our mini biosystems. These are small glass autoclavable bioreactors that has the same characteristics as the larger lab scale systems. These systems are available in multi-use autoclavable systems, but also in single-use systems to ease the workflow. Next to these mini biosystems, we also have for typical lab scale systems. These are 2 to 20 liter glass autoclavable bioreactors. Now we go to the point where we go into the pilot production systems. Here we can supply stainless steel systems that still can fit on a bench or are working standalone, and that you have steam in place and clean in place functionality. We also offer, in cooperation with Thermo Fisher Scientific, we offer their high performance systems with an Applicant sub-controller. This allows for single-use solutions up to 2,000 liter working volume. Looking into full cGMP production facilities, we can supply custom projects with custom software packages that meet the requirements for pharmaceutical industry. So Optical Biotechnology has all the equipment to offer, from research on the lab scale to full cGMP production. So we've now seen the full Applicant product line. Today, we will focus on the single-use portion of our laboratory scale bioreactors. That are the OptiFlex ST single-use bioreactors. What we're going to do is we're going to have a look at a short introduction on that. We're going to have a look at the novelties that we have, and we're going to be closing off with an application where these bioreactors are used in the field for a cell therapy application. But now first, we're going to start off with a short introduction. The OptiFlex ST is a fully customizable bioreactor. We do that by 3D printing most of the components of the bioreactor. And this allows us to have full flexibility on how we design the bioreactor. This means we can really make a bioreactor dedicated to your process or to your cell line. As you can see in the 4 images that you see here, this gives a good example of the flexibility that we have with 3D printing. We can customize the impeller of the bioreactor, but also the head plate and the fittings. Being able to customize the fittings that are on the bioreactor also allows us to customize the tubing kit that goes on the bioreactor. This means tubing type, tubing length, which connectors go on there. This to ensure that your bioreactor connects seamlessly into your process. Next to the impeller and the head plate fittings, we can also customize addition ports, sample tubes, spargers, etc. This gives us full flexibility. The OptiFlex ST single-use bioreactor has also been designed to be able to switch between a multi-use and a single-use system. This means that with a small modification on the hardware of your glass autoclavable system, you're able to use the OptiFlex ST single-use bioreactor as well on that same system. This prevents you from doing a large capital investment if you want to switch to single-use bioprocessing. The most commonly used bioreactor size for the lab is a 3-liter bioreactor. And here you see the 3-liter OptiFlex with the 2 control tower options that we can offer. The left is the MyControl control tower, which you access via a web user interface, and the right is the EZ2 control tower, which you're able to access via the web interface or via the screen that is on top of it. Now that we've seen what the OptiFlex ST product is, we're going to have a look at the novelties. We'll be discussing single-use sensors, a microbial version, and a bioreactor for perfusion applications. Once we've done that, we'll have a look at a cell therapy application from one of our users. But first, we're going to have a look at the single-use sensors. With a glass autoclavable bioreactor, you always need to calibrate your sensors prior to the sterilization of the bioreactor before you are able to start your run. We are now introducing single-use sensors into the OptiFlex ST, which makes that a thing of the past. The new OptiFlex ST models come with pre-calibrated sensor dots. This allows the user to just insert the calibration data into the bioreactor control system, and then we're ready to start. We will have a look later into the detailed procedure for this calibration routine. As you can see, in the bottom of the vessel, optical spots are placed. These are the measurement spots for pH and dissolved oxygen. Via optical cables, these are connected via a special readout module, which is then connected to the controller. Now, let's have a look at the procedure to calibrate the sensors and the sensor specifications. So we're now going to have a look at how to set up the single-use pH in the DO sensor. For this, we're now using the web UI, but we could also use the main embedded interface on the Easy Control. So there are a couple of steps we need to do. We need to insert for the pH sensor the calibration data, and we need to do an offset correction. During this, we need to have sufficient time for the sensor signal to stabilize in order for us to do a decent offset calibration. So let's get started. First, we're going to be logging in as an operator. And then we can navigate to the pH sensor, to the settings. And here we have the phases and the other calibration data. So what we're going to do here is we're going to put in the calibration data that comes with the box of OptiFlex STs. So we can see here, this needs to be this number, and we can agree on that. This needs to be 30.26. This needs to be 5.24. And this needs to be 0.53. And we see that the calibration has been done at 37 degrees, so we also have to insert 37 degrees here. All right, so now the settings are in. You can check that. Yes, correct settings are in here, and then we need to calibrate. Of course, this is a visualization, not an actual measurement, but say the actual sample value we needed to be 7.1, and we type 7.1 here, and then we need to calibrate the correction, and then we go, and it is calibrated. So with this, we can do the offset calibration. Of course, doing the offset calibration, we do need to allow the pH sensor for enough time to stabilize, but once that is done, you're now ready to start the pH loop. So now we're going to have a look at how to calibrate the dissolved oxygen single-use sensor. First, we are logged in as an operator, so we don't need to do anything there, and we can navigate to the DO2 sensor. So here we find settings, custom, and here we find the same fill-in fields as we are used to for the pH sensor. So also here we fill in the correct data. The temperature is still in the same range that it needs to be, or the same value. This is 24.77. And now we have inserted all the data from the calibration sheet into the controller. So now we're basically done. The sensor has been calibrated. Only we do advise to do another calibration with this to calibrate for a 100% value. Once you have calibrated the pH in the DO sensor using the methods that we have just seen, you'll be able to get the following range. The pH sensor works in a range from 6 to 8 with an accuracy of 0.1 pH points and a response time from less than 60 seconds. We do recommend, in comparison to the traditional pH sensor, to lower the proportional gain of the PID setting to 20. And during a cultivation, when you do a large pH step, like say more than half a pH point, please redo the offset calibration. And the accuracy that we mentioned, we state that that is obtained after sufficient stabilization time, as mentioned here. For the DO sensor, you can use the default PID settings, and the range and accuracy there are given. The key advantage of the single-use sensors is the time saved setting up and preparing the bioreactor, and the risk mitigated in opening up the bioreactor for sensor insertion. Thus, the single-use sensors allow for a smoother workflow overall. With that, we conclude the section on the single-use sensors. The single-use sensors in the OptiFlex ST really allow you to do a box-to-bench approach. You take the bioreactors from the box into the bench, and you're ready to start your cultivation right away. Continuing, we're going to have a look at the perfusion solutions that we now offer in the single-use product line. This is our acoustic separation system, the BioSep. First, we'll have a look into an introduction on the BioSep and how it works, and then we see how we've implemented that into the single-use OptiFlex SG. Let's discuss the principle of operation of the BioSep first. In this image, you see the BioSep included in a bioreactor setup. In the BioSep, cells are separated from the media. This is done by acoustic standing wave technology. We'll have a look at this in a bit. What we do is that we have a recirculation flow going into the BioSep chamber, this image coming in from the right, and then exiting below. A part of that flow is exited the bioreactor system via the harvest. And then in the BioSep chamber itself, the cell separation takes place. Of course, when we do the harvest, we also need to add fresh feed to the system. The BioSep operates with acoustic standing wave technology. An acoustic wave is sent through the BioSep chamber, and that creates sections with low pressure zones and high pressure zones. And then the cells accumulate in the low pressure zones. You can see this happening in the following image. You see that cell bands are created where we have a high concentration of cells. Locally, the cell density is higher there, and thus the cells will drop out of suspension. At the top of the system, the harvest flow is withdrawn, and slowly cell-free media is exited there. What you see here is that this is not an absolute filter, such as an ATF or a TTF membrane, but this is an acoustic filter. Here we see a detail of how that cell banding looks, and the red arrows indicate the flow of the media, which is the arrows above, and then the flow of the cells. So this is the principle of operation for the BioSep system. Next up is the OptiFlex ST 500 ml for a reusable BioSep for cell applications. What we've done is we've created a custom OptiFlex ST design for the BioSep, and we made that into one of our default configurations. This means that you could order this OptiFlex ST model off the shelf at Optical Biotechnology without going into a customization project. Well, as you can see on the head plate here, there are quite some additions in comparison to a normal cell configuration. For example, we have got level sensors to measure the level inside the bioreactor. We've got an additional sensor port, and we've got a bent dip tube that goes to the BioSep. So let's have a look in a bit more detail at the configuration. The BioSep itself has also been updated. or to be more specifically, the tube which sits into the bioreactor that connects the biosep has been changed. It has been changed from a form on which it has been a straight pipe to a form where it has a small bend. On the right side here, you can see how that fits into the bioreactor if all the sensors are placed. Combining all components into one single system results in a system that looks like this. Here you see in the center the Optiflex ST with the biosep and all the sensors and motors mounted, plus 2 external pumps and the biosep controller. That concludes the session on the single-use sensor versions of the Optiflex ST. We're now going to have a look at the latest model out, a 3-liter model for microbial processing. We've developed a 3-liter bioreactor for microbial applications in a single-use format. This bioreactor consists of 2 Rushton impellers for microbial application. Also, an internal heat exchanger is placed to get rid of excess heat. There are 2 versions available. There is one high-density version able to meet the high KLA requirements that a high-density culture requests, and that contains a stainless steel heat exchanger, and we have the regular version that has a tube heat exchanger. These models are now available. We've now had a look at the novelties in the Optiflex product lines. We're now going to have a look at an application example. Together with Delft University and Sanquin, the blood bank of the Netherlands, the Optiflex ST has been used to cultivate red blood cells. We've invited Joan Gallego to this talk to give you an introduction into the project and to share some of the results here. Without further ado, I'd like to give the floor to Joan.
Hello everyone. Thanks for joining this webinar. I am Joan Gallego, third-year PhD student in the Department of Biotechnology at TU Delft and in the Hematopoiesis group at Sanquin, the Blood Supply Institute of the Netherlands. Today, I will be sharing with you some of our results on the upscaling of the production of red blood cells for transfusion purposes. So let's start by talking about blood transfusion. Blood transfusions is the most common cell therapy to this date. In 2018, more than 100 million blood donations were collected worldwide. That same year, 724,000 donations were collected in the Netherlands by Sanquin, the National Blood Bank. From these, 404,000 red blood cell concentrates were obtained and supplied for transfusion purposes. The current system is fully donor dependent. This means that the only way to obtain red blood cells for transfusion purposes is obtaining it from donors. There are several challenges in this fully donor-dependent system. For example, even though blood obtained from donors is screened for blood-borne diseases, the cost of screening for such diseases or for novel ones is high. More important, it can be difficult to obtain red blood cells with the specific blood groups that are needed by some patients. This is aggravated when those patients need recurrent transfusions and they start to develop alloimmunization. One way of tackling these issues is by culturing cells for transfusion purposes. This way, we could, in theory, produce red blood cells on demand, especially when the current donor-dependent systems fall short, have more control on the quality and safety of the product, and we could have a final product that is compatible for people with special requirements in terms of blood groups. Not only we could produce plain or raw red blood cells, but we could also produce red blood cells loaded with therapeutics, which could be then used as therapy for patients with specific diseases. However, before we are able to use cultured red blood cells, we need to produce them first, and produce them in large amounts. So how do we culture red blood cells in our labs? We normally start from hematopoietic stem cells that are cultured for 6 to 8 days in conditions that push them into the erythroid lineage. This first stage is known as the progenitor expansion phase. At the end of this pre-culture phase, we have proerythroblasts, which are the precursors of the red blood cells. These erythroblasts are cultured in expansion media in which they proliferate extensively. This phase is called erythroblast expansion. When we have enough of those cells, we change the media composition and add specific growth factors that push the cells into a differentiation process. In this differentiation stage, erythroblasts mature into red blood cells that can then be used for transfusion purposes. They also stop growing in this stage, dividing only 1 to 3 times more. For this presentation, we will focus on the erythroblast expansion stage. This is because the main challenge of any culture red blood cell protocol is that we need to produce large amount of cells, as a single unit of packed red blood cells for transfusion purposes contains 2 trillion cells. For our protocol, that represents large culture volumes, in the order of 200 liters for both expansion and differentiation stages, due to the low cell concentrations that need to be maintained. The large volume requirements, of course, lead to high operating costs, and also to extremely large cultivation areas, in the order of 20,000 culture dishes for a single transfusion unit. In summary, there are 3 main biological requirements that we need to consider when upscaling this process. First, we need to consider the conditions in which these cells are growing in our body. Erythroblasts proliferate in the bone marrow, where there is a low to no shear stress, and where there is a controlled supply of oxygen. We also need to deliver fresh nutrients and growth factors to the cells to keep them proliferating, at the same time that we remove potential inhibitors that could be produced during growth. These biological requirements translate to 3 main challenges that need to be tackled to upscale this process: the inconvenient method of cultivation that we use at the moment, the media cost, and the limitations in cell density. Thus, our aim, and specifically the aim of my PhD project, is to develop strategies that could allow us to tackle these challenges to make the production of red blood cells something feasible. Currently, we produce cultured red blood cells in culture dishes, which are static culture conditions. In these kind of static systems, we have multiple problems. First, we have gas transfer limitations. We also have gradings of nutrients, as cells tend to be located only at the bottom of the dish. And finally, when you scale this process up, you're scaling up by area, and working with so many dishes is cumbersome and time-consuming. One way to tackle these challenges is to move to suspension systems like stirred tank bioreactors. As you know, in a stirred tank bioreactor, we could keep our culture fully homogeneous, at least that is what we normally aim to, while having full control over important variables such as pH and oxygen concentration. In comparison to culture dishes, this type of systems is scaled up by volume. The idea of using stirred tank bioreactors for culture red blood cell production is being explored by other groups, but then when they go to larger scales, they still face some of the challenges I mentioned before. One of the main challenges when you start testing cell cultures in bioreactors is that each one of these bioreactor runs can be expensive, because now you're using large medium volumes, and if the reactor fails, as it often happens when you're optimizing growth conditions, it takes a significant time to start again due to the slow pre-culture process. Because of this, we decided to use a bioreactor platform that would allow us to have low culture volumes. Specifically, we use the MiniBio 500 milliliter bioreactors from Optical Biotechnology. These are autoclavable reactors in which we can have cultures from 100 to 400 milliliters with full control on pH, dissolved oxygen, and temperature. Since already some years, there has been a drive to implement single-use bioreactors in pharma. Specifically, in our case, we're interested in having a system that can be easily translated to good manufacturing practices expectations, as the cells produced in the reactor will be then used in patients. Thus, we decided to use the Appliflex ST 0.5 liter bioreactors, also from Applicant. One of the advantages of using single-use bioreactors is that they come fully assembled, meaning that it saves some time when starting new cultivations. Also, the Appliflex platform is based on 3D printing technology. It is straightforward to customize them. For example, multiple impeller geometries can be designed and used. This option for customization opens new possibilities to have more control on, for example, the turbulence and circulation patterns in the reactor. This is important because, as some of you probably know, mammalian cells are known to be less tolerant to turbulence and shear stress compared to, for example, yeast and bacteria. Due to the importance of turbulence and shear in mammalian cell cultures, we decided to quantify these variables in the Appliflex 0.5 liter and the MiniBio 500 milliliter bioreactors, to know to which values our cells will be exposed in these bioreactors. To do this, we perform computational fluid dynamic simulations of the culture. We assume a single phase, only the liquid, to only quantify the effect of mixing. From our CFD simulations, we could then estimate the shear rate and stress at any point of the reactor. We could also calculate the turbulent energy dissipation rate, or EDR, which can be interpreted as the local power per volume input. From those results, we can make plots like this histogram in which we could see the fraction of the cultures exposed to specific values of turbulence. As you can see here, most of the reactor volumes have a local EDR lower to 10 watts per cubic meter. You can imagine that at very high EDR values, cells will be exposed to very high turbulence, which could lead to negative effects on growth or even cell death. This is exactly what has been described in literature. In this plot, the lethal range of EDR is highlighted in red, while the so-called sublethal effects, such as low growth rate or lower product yield, takes place in the yellow region. From our simulations, we could observe that the EDR values in the Appliflex bioreactors are well below the lethal and sublethal regions, suggesting that the mixing in this type of reactor should be okay for our cultures. After these simulations, the first thing that we tried was to check if we could implement our erythroblast expansion protocol in this type of bioreactors. For this, we started with our conventional protocol, starting from hematopoietic stem cells, and we cultured them in dishes for 12 days until we had enough erythroblasts to inoculate either the mini-bio or Appliflex reactors at a concentration of 1 million cells per milliliter. The bioreactors were operated in what is known as repeated batch mode, which is nothing else than mimicking what we do in our culture dishes. We start from a low cell concentration, we leave the cells growing, usually overnight, and then we remove some of the culture and replace it with fresh media, resetting again the cell concentration to a low value, while giving the cells fresh nutrients and growth factors to proliferate further. One of the main challenges in bioreactor cultures, apart from the low throughput of experiments you can do, because you need a lot of cells and multiple bioreactors to run things in parallel, is that there are always differences in the inoculum used for the cultivation. Even though the reactor is always inoculated with day 12 cells, donor differences or even differences during pre-culture if we use the same donor lead to variations in the growth curves. Still, we could observe that all the bioreactor runs, here indicated by the different curves in different colors at the left, show similar growth to what is observed in the culture dishes, shown in the right. Here we have a single run in which we compare the two bioreactor systems, the mini-bio and the Appliflex, and the static conditions. We observe that in the bioreactors we have similar growth profiles compared to what we obtain in our controls, which are the static culture dishes. We also had high viabilities in the bioreactor cultures, at least comparable to what we observe in the dishes. This led us to conclude that we could indeed sustain erythroblast proliferation in the tested bioreactor setups, and that the results in the autoclavable mini-bio are comparable to the single-use Appliflex bioreactor. An important parameter that we have to keep under control in our cultures is the differentiation stage of the cells. This is because we want to have proliferating cells in this expansion stage. If the cells start to differentiate, they also lose some of their proliferation potential. And this is very difficult to control, mainly because it's still not known specifically what causes this spontaneous differentiation. One way to check if the cells are differentiating more or less is by checking the expression of protein markers in the surface of the cell. Two typical markers are CD235 here in the x-axis and CD71 here in the y-axis. If you're not familiar with flow cytometry results, in this plot, each dot is an individual cell that has been stained for these two markers. So yes, there is heterogeneity in the cultures. However, as a whole, if a cell population is differentiating, it moves to the double positive corner, and then it starts to move down following kind of a waterfall shape. We can observe that there is some spontaneous differentiation happening in the bioreactor, shown here by the culture moving to the double positive CD71, CD235 gate, and then to the CD235A positive, CD71 negative corner. However, it is again comparable to what we observe in the dishes. In this figure, I only show the percentages of the 4 corners or gates. More differentiation would be shown as a larger dark color bar. As I told you, reactors and culture in static conditions look very similar. With this, we could conclude that indeed we could expand erythroblasts in the certain bioreactor without a negative effect on the differentiation state of the cells. As I mentioned at the beginning, one of the main advantages of a stirred bioreactor is that it's really easy to measure and control culture variables, such as oxygen concentration. Of course, this is something that is natural for us because we work on or we know about bioreactor cultivations, but it's not trivial when you consider that the small-scale animal cell cultures in research are done in incubators where there is no measurement or control of oxygen levels in the liquid. From the first bioreactor experiment we performed, we could also observe that oxygen control is essential. Here in the left, I'm showing you the evolution of oxygen concentration during the bioreactor culture for 2 bioreactor runs. 100% is equivalent to having the culture full saturated with oxygen, which is equivalent to roughly 6.1 micrograms of oxygen per milliliter for these conditions. For the light blue-green reactor, we didn't have any oxygen control, and we observed that the oxygen concentration went to really low values, less than 1% of saturation, after 6 hours of culture. This led to a decrease of the viability of the culture in the following days. For the pulper reactor, we controlled the oxygen level by sparging air when the oxygen concentration level went below 40% of saturation. We chose 40% because it is a typical value used in other mammalian cell cultures. You can see the effect of this intermittent sparging here in the many small peaks of oxygen concentration profile. For this reactor, we didn't see that drop in viability at all. So we know now that some sparging is needed for the culture. This intermittent sparging strategy has shown to be a good approach, as continuous sparging leads to too much foam, which we initially thought it could affect negatively the growth of the cells, at least based on what is reported in literature. Another advantage of having the continuous measurement of oxygen concentration in the bioreactor is that we can use this data to calculate the oxygen requirements of these cells. From our data, we could calculate that the erythroblast in expansion require between 0.5 and 3 picograms of oxygen per cell per hour. This value is comparable to what has been reported for other mammalian cell cultures. As it is relatively easy to control oxygen concentration in the reactor, we wanted to know if we could expand erythroblasts at low oxygen concentrations. For this, we chose 2 set points, 10% and 40% of the oxygen saturation concentration. This is relevant because these cells are normally growing in the bone marrow in our bodies, where the oxygen concentration can be 5 to 10 times lower than what I showed you before as 100% saturation. Here in the left, I'm showing you the oxygen concentration and how it is being kept around those values during the whole bioreactor cultivation. In terms of growth, there seems to be no difference between the 2 conditions. This is also true for the differentiation level of the cells. And also in terms of the viability, here shown with the apoptosis marker annexin-5 and the live death marker DRAG7. But why evaluating if 10% is okay is relevant? Well, most of you know that oxygen transfer rate is proportional to the gradient in oxygen concentration between the gas phase, which is the saturation concentration, and the liquid phase, which is the concentration of oxygen in the culture. If you have a big differences in concentration, then the transfer is faster or more efficient. This is relevant because if we are able to grow these cells at 10% of saturation, as we showed here, we can have a faster, more efficient oxygen transfer than a higher set point like 40%. This would mean that we would need less sparging to keep the oxygen concentration at the levels that we want. Nevertheless, we need to consider that going low in dissolved oxygen set points can be a potential problem, as in large scales it is more difficult to keep everything well mixed, and it would be easier to have regions with oxygen concentrations close to 0. With this, I would like to finish this presentation by going back to the biological requirements that I showed you in one of the first slides. Our results show that some of these challenges can be tackled, putting us closer to the production of red blood cells for transfusion purposes. Regarding the method of cultivation, we show you that we could maintain the expansion of erythroblasts in stirred tank bioreactors, specifically the Mini-Bio 0.5 liters and the Appliflex 500 milliliter bioreactors for at least 6 days. This was achieved by controlling oxygen levels in the reactor and by choosing in a smart way the geometry of the impeller to have control on the shear stress and turbulence to which cells would be exposed. We're currently working on scaling up further the cultivation by testing the Appliflex 3-liter bioreactors. The cost of the medium and the cell density limitations were not addressed in this presentation, but both are things that we are investigating now. For the cost of the medium, we're trying to reduce and replace the expensive components in the medium. And to tackle the cell density limitations, we are also evaluating a perfusion feeding approach to increase the cell concentration inside the reactor while feeding fresh nutrients to the cells and removing potential inhibitors that are accumulated in batch or fed batch operating modes.
With that, we want to conclude also this presentation. We're very happy that Joanne was able to present his work here. If there is any feedback or comments or questions on the presentation or the content, feel free to reach out to us and we'll be able to answer those questions. You can also use the chat now and we'll be able to look at that. If there are no further questions, I want to thank you for your time and we'll be talking the next time. Thank you.
Tom, thank you very much for this nice presentation. Also with the application example, thank you.
Thank you, Berna.
As far as I see, there are no questions coming from the chat window. We have 1 minute left, so maybe they can reach us later if they have any questions.
Yeah, that's not an issue. If there are any questions on the topic, then feel free to reach out to On Technique, and they'll be able to touch base with myself. But also, if you have questions for Joanne, we'll be able to forward those and get you guys into contact.
Okay. I guess we can leave. It's last 50 seconds. So thank you all for joining.
Thank you too, and hope to see you again on the next seminar, maybe even face to face the next time.
Yeah, hopefully. Okay, see you.
All right.
See you later.
Hepimiz için güzel geçsin. Dileklerimizle başlıyoruz. Öncelikli olarak bugün sizlere BioExpo kapsamında Ali arkadaşımla birlikte regülasyonlara göre filtre validasyonunda ürüne uygun doğru filtrenin seçiminin öneminden bahsedeceğiz. Ben uzman biyolog Erol Aydın, sarkonet separasyon teknolojilerinde 15 yıldır çalışıyorum. Özellikle satış müdürü ve son 3 yıldır filtre validasyon projeleri alanında çalışıyoruz. Üniversite olarak 19 Mayıs, Samsun 19 Mayıs Biyoloji bölümünden mezun oldum. Yüksek lisansımı Marmara Üniversitesi'nde tamamladım. Buradan önce yaklaşık 4,5 yıl bir ilaç firmasında, veteriner ilaç firmasında bölüm sorumlusu olarak çalıştım. Evet Ali, sen de istersen kısaca bahset kendinden.
Ben Ali Rahmi Fırat, ben de biyoloji bölümü mezunuyum. Yine lisansüstü eğitimlerimi Çanakkale 18 Mart Üniversitesi'nde devam ettim. lisansüstü eğitimlerim boyunca çeşitli TÜBİTAK projelerinde ve Orman ve Su İşleri Bakanlığı projelerinde yer aldım. Aynı zamanda yine eğitim sürecim boyunca ve eğitimlerimin sonrasında da bilimsel araştırmalarla hem yurt içinde hem yurt dışında yaptığımız yayınlarla bilimsel çalışmalarıma devam ettim. Şu an için yaklaşık olarak 4 yıldır Sartonet ailesiyle beraber çalışmaktayım. Sartonet'e katıldığımdan beri filtre validasyonu kapsamında Türkiye'deki filtre validasyon laboratuvarımızda hizmet vermekteyim. Laboratuvar sorumluluğunu yürütmekteyim. Bugünkü sunumumuzda Erol Bey ile beraber sunumu tamamlayacağız. Erol Bey yine regülasyonun söylemiş olduğu filtre validasyonu hakkındaki bilgileri bize verirken ben de teknik olarak konuyla ilgili detayları sizlere sunacağım.
Evet. Bu süreçte uygulamalı olarak neler yaptık, ne tür çalışmalar yaptık, o konuda teknik olarak Ali arkadaşım sizlere bilgiler verecek. Ben kısaca Sartorius firmasından bahsedeyim, sizlere bilgi vereyim. Sartorius 150 yıllık bir firma, Alman menşeli bir firma ve bugün günümüzde birçok alanda, ilaç endüstrisinde, mikrobiyolojik analizlerde, damara enjekte edilen ürünlerin filtrasyonunda kullanılan membran filtrenin mucididir Sartorius. Aynı zamanda dünyaca ünlü hassas terazileriyle bilinir ki bir tükenmez kalemin kağıt üzerinde bırakmış olduğu mürekkebin ağırlığını ölçebilecek hassas terazileriyle meşhurdur. Sartonet separasyon teknolojileri olarak da yaklaşık 40 yıldır Türkiye'de Sartorius firmasının temsilciliğini yapmaktayız. Bu kısa bilgiden sonra özellikle bugünkü konumuz regülasyonlara göre filtre validasyonunda ürüne uygun doğru filtrenin seçiminin önemi olacak. İsterseniz sunumumuza hızlıca başlayalım. Evet, konu başlığımızı söyledik. Regülasyonlara göre filtre validasyonunda ürüne uygun doğru filtre seçiminin önemi konu başlığımız. Ülkemiz 1 Ocak 2018 tarihinde ilaç camiası için çok önemli olan Uluslararası İlaç Denetim Birliği üyeliğine kabul edildi. Üyelik için başvuru tarihini tahmin edebilir misiniz? Ben bu konuda size bir bilgi vereyim. Yaklaşık 30 yıl öncesinde böyle bir başvuru süreci başlıyor ve bu süreç içerisinde gerekli altyapı çalışmaları Sağlık Bakanlığımız ve TİCK tarafından tamamlanarak 1 Ocak 2018 tarihinde ilaç camiası için çok önemli olan, ülkemiz için çok önemli olan PIX üyeliğine kabul ediliyor. Bu PIX içerisinde hangi ülkeler var diye baktığımızda ilaç endüstrisinde en ileride olan, en gelişmiş teknolojilere sahip olan başta Almanya gibi, Amerika, İngiltere, Kanada, Japonya gibi dünya ilaç sektörünün en önde gelen ülkeleri yer almaktadır. Üyelik neden önemlidir? Bu üyelik ile Sağlık Bakanlığımızca Türkiye İlaç ve Tıbbi Cihaz Kurumunca yapılan tüm denetimler dünyada kabul gördü. Ülkemizde üretilen ilaçların kalitesi ve güvenilirliği tescil edildi. Yurt dışına ilaç ihracatının yani dünya pazarlarına açılmanın önündeki teknik engeller de ortadan kalkmış oldu. Geçtiğimiz sene yine çok önemli ilaç camiası için önemli olarak bilinen ICH dediğimiz yani Uluslararası Uyum Konseyi'ne de tam üye olarak Sağlık Bakanlığımız, ülkemiz kabul edildi. TİCK'nın çalışmaları neticesinde. Türkiye bundan sonraki süreçte dünyadaki ilaç geliştirme ve üretim süreçlerinde aktif rol oynayabilecek. Sağlık Bakanlığımız yani TİCK ilaç konusunda sayılı otoriteler arasında yerini almış oldu. Yine TİCK Uluslararası Uyum Konseyi'nin teknik kılavuzlarına uyum sağlamakla kalmayıp söz konusu kılavuzların oluşturulmasına da katkı sağlayacak bu üyelikle birlikte. ICH için yayınlamış olduğu birçok kılavuzlar var. ICH Uluslararası Uyum Konseyi dediğimiz International Council for Harmonization'ın birçok yayınlamış olduğu alt kılavuzlar var. Bunların hepsi ilaç camiasında takip edilen, ilaç camiasının gelişmesine destek olan kılavuzlardır. Özellikle bu kılavuzlardan ICH Q8'de filtre seçiminde Calibrate the Design olarak da bizim söyleyebileceğimiz günümüz ilaç otoritesinin farmasötik ürünlere bakış açısını yansıtan en önemli konseptlerden bir tanesidir. İlaç sanayinde kalitenin kurallarını belirleyen iyi üretim uygulamaları yani GMP uyum için ürünün üretim ömrü boyunca, yani gittikçe daha fazla sayıda test yapmaktansa kaliteyi planlayarak garanti altına almak gerektiği fikri son yıllarda ağırlık kazanmıştır ve bu fikir ICH Q8'de şöyle ifade edilmiştir: bitmiş bir ürüne test yapılarak kalite kazandırılamaz. Kalite tasarımla kazandırılmalıdır. Peki bu PIX ve ICH üyeliği neler getirdi? İlaç üretiminde kalite ve güvenilirlik için belli standartları da tabii ki zorunluluk haline getirmiştir. Bunlardan bir tanesi de filtre validasyonudur. Bu zorunluluk likit ve non-steril, likit olarak bildiğimiz steril ve non-steril ilaç üretimlerinde kullanılan filtrenin validasyonudur. Peki filtre validasyonu nedir? Prosese ve ürüne uygun doğru filtrenin seçimiyle başlar filtre validasyonu. Bu ilk basamağıdır. Fakat diğer ilerleyen basamaklara direkt etki edeceği için en önemli basamağıdır. Burada yapılacak olan bir hata, bir yanlış uygulama diğer tarafların da hatalı çıkmasına yol açacak. Gerek maddi gerekse zaman kaybına yol açacaktır. Bir diğer kısım seçilen filtreye ait kalite belgelerinin dokümante edilmesidir. Yani kalifikasyon olarak adlandırdığımız kısmıdır. Ve son olarak da işte filtre validasyon testleri olarak adlandırdığımız proses şartlarında ve gerçek ürün varlığında filtrenin ürün ile kimyasal uyumluluğunun tespiti, yani chemical compatibility olarak adlandırdığımız test, kendisinden, yani filtrenin, seçilmiş olan filtrenin kendisinden beklenen görevi yapıp yapmadığının kontrolü, yani aseptik filtrasyonu yapıp yapmadığının kontrolü, ürün içeriğini etkilemediğinin, yani kendi yapısından ürüne herhangi bir kimyasal bırakmadığının ve ürünümün içeriğindeki herhangi bir etken gibi, diğer yan bileşenler gibi, taşıyıcılar, prezervanlar gibi bütün bileşenlerinden herhangi bir şeyi de üzerinde tutmadığının gösterilmesidir. Ve bunların tabii adsorpsiyon ve işte extractable, leachable olarak çeşitli adları vardır. Bunların her birisi ayrı ayrı çok detaylı konulardır. Biz özellikle 3 yıldır ülkemiz PIX üyesi ve ICH üyeliğiyle birlikte bu tabii biraz daha önem kazandığı için son 3 yıldır özellikle sartonet separasyon teknolojileri olarak bu konudaki 40 yıllık bilgimizi bütün ilaç camiasındaki paydaşlarımızla zaman zaman yaptığımız online sunumlarla, ki bazılarında da ilaç firmaları özellikle bu konuda bizden talepte bulundular. Onlara giderek firma bazında eğitimlerle bildiğimiz bilgileri anlatıyoruz. Özellikle filtre validasyonunu bir piramide benzetecek olursak, bir piramidin en güvenli, en sağlam kısmı en alt kısmıdır tabii ki. Ve bu da işte proses ve ürüne uygun doğru filtrenin seçimi olarak biz bunu adlandırıyoruz. Diğer taraftan kalifikasyon ve validasyona gittiğimizde de validasyon testlerini görebiliyoruz.
Burada Erol Bey, müsaadenizle bir şey eklemek istiyorum. Burada doğru filtre seçiminde aslında daha sürecin başlangıcında riskler minimize edilerek simülasyon, laboratuvar ölçekli çalışmalarla doğru materyal seçilir ve prosesteki verimlilik arttırılır. Yani ilerleyen süreçte yanlış materyal seçildiyse validasyon süreci daha tamamlanamayacağından ötürü sürecin başa dönmesini bu noktada engelleyip efektif bir süreç başlangıcı yaratılmış olur. Regülasyon da zaten kayıpların önlenmesi için laboratuvar ölçekli bu tarz simülasyon denemelerinin ve doğru filtre seçimi çalışmalarının yapılmasını önermektedir.
Teşekkürler Ali, katkın için. Dolayısıyla biz aslında filtre validasyonu olarak bunu bütünsel olarak söylüyoruz ve bunu da bir piramitteki işte ilk basamağı ürüne uygun doğru filtrenin seçimi, ki bugünkü temel konumuz bu. Diğerleri başka sunumlar konusu olarak karşımıza çıkıyor. Burada özellikle benim aktaracağım kısımda filtre validasyonu ile ilgili ne tür regülasyonlar, hangi tür bilgilerle karşımıza çıkıyor onu gösteriyoruz. Seçeceğim filtreden beklentilerim nelerdir? Burada bu beklentileri aslında regülasyon, özellikle EMA guideline'ında görebiliyoruz. Burada dikkatli bakacak olursak seçeceğim filtreden beklentim gözeneklerinden daha büyük partikül ve mikroorganizmaları %100 tutması, yani aseptik filtrasyonu yapması, en küçük boy filtre ile en kısa sürede en fazla ürünü süzebiliyor olması, kullanıcı tarafından da test edilebiliyor olması, herhangi bir maddeyi tutmaması ve herhangi bir maddeyi de kendi yapısından bırakmaması. Bu sol taraftakileri biz aslında formülize ettik ama yan tarafta EMA guideline'ına baktığımızda birebir bunları bizden istiyor ve özellikle de burada sadece beşeri ürünlerde değil, veteriner ürünleri de kapsayacak şekilde, oradaki kullanılan filtreleri de kapsayacak şekilde aynı özellikleri aslında istiyor EMA Guideline. Ve burada istenen özellikle ürünle bu çalışmaların yapılması. Yoksa bu çalışmaları zaten filtre tedarikçi firmalar su ile veya çeşitli solventlerle kendisi filtre üretme aşamasında bunları zaten yapıyor. Ancak PIX üyeliğiyle birlikte karşımıza çıkan sizin ürününüzle birlikte bu testlerin yapılması ve bunun ilgili otoritelere gösterilmesidir. Peki hangi otoriteler dedik, regülasyonlar diyoruz, hangileri? Tabii ki en başta Sağlık Bakanlığımız. Sağlık Bakanlığımızın bir GMP kılavuzu var. İşte o da zaten Avrupa Birliği çerçevesinde Avrupa GMP'sinden teknik tercümelerle, çevirilerle oluşturulmuştur. Diğer taraftan işte PIX dedik. ISO'nun bu konuda özellikle ilaç endüstrisine yönelik üretilen ürünlere yönelik bilgi kılavuzları var. ICH'lik, FDA'nin yine aynı şekilde yine PDA Teknik Report 26 dediğimiz çok detaylı bir şekilde aseptik filtrasyonla ilgili tanımlamalar, açıklamalar var. Japon Farmakopisi, ISPE gibi otoriteler aslında aynı konuları birbirinden çok ufak farklılıklarla değişik maddelerde ele almaktadır. Örnek verecek olursak, Ödrelex burada filtrasyon sistemi bileşen seçimi için hava ve gaz havalandırmaları için de bunu istiyor. Filtreler dahil, burada tabii sadece filtreleri de istemiyor. İlaç üretiminde kullanılan bütün ekipmanların mutlak surette bir ön değerlendirmeye tabi tutulmasını istiyor. Filtreleme sistemi içindeki ara bağlantılar, contalar, silikon olabilir, diğer materyaller de olabilir. Ön filtreler de dahil ürünlerin kritik kalite özelliklerine dayanmalı, belgelenmeli ve doğrulanmalıdır şeklinde buradaki 8.79. maddesinde Ödrelex bunu ifade ediyor. Yine ISO 134.08-2'de sağlık ürünlerinin aseptik olarak işlenmesi filtrasyon kısmında filtre kullanıcılarının bilgilerine dayanan sıvıya özgü seçim kriterleri maddesi var. 6.1'de filtre kullanıcısı filtrasyon prosesini ve filtrelenecek sıvıyı dikkate alan dokümante edilmiş bir filtre değerlendirme programı takip edilerek filtrenin karakterini değerlendirmelidir diyor. Yani burada bakın bir filtre değerlendirme programından bahsediyor. Dolayısıyla biz bir üründe filtre kullanacağız. Bunu bir değerlendirme programından geçirerek seçmeliyiz, ele almalıyız şeklinde ve bu filtreyi yine devamında regülasyonun bakın birebir çevirisidir bu. Biz bu regülasyona herhangi bir şey ekleme veya çıkarma yapma şansımız yoktur. Regülasyon ne diyorsa bunu aynı şekilde aldık. Filtrenin karakteri filtrelenecek sıvıdan olumsuz bir şekilde etkilenmemelidir. Aynı zamanda ürün de filtreden olumsuz bir şekilde etkilenmemelidir. Sıvı bileşenlerin absorpsiyonu ve filtre bileşenlerin ekstraksiyonu değerlendirilmelidir diyor. Ve bunu yaparken prefiltreleri de göz önünde bulundurarak bu çalışmaları prefiltrelerde de istemektedir. Yine PIX validasyon ana planı ile ilgili tavsiyelerde 6.3.1 maddesinde ürün geliştirme sürecinde üretim prosesi tek tek basamaklara bölünmelidir. Her basamak bitmiş ürünün kalitesini etkileyebilecek kritik faktörler, parametreler teorik ve tecrübeye dayalı olarak belirlenmelidir diyor. Yine bir diğer maddesinde bakın bu faktörlerin kritikliğini belirlemek için bir seri deneyler kurgulanmalıdır. Yani bir filtreye karar vereceksiniz bir ürününüzde. Dolayısıyla bir filtre programını takip edeceksiniz ve bazı testler yapacaksınız bu filtreyi seçerken. Şimdi A filtresini seçerken, B filtresini seçerken farklı deneyler kurgulamalısınız diyor. Ve bir ilaç firmasını düşünecek olursak üretim, kalite kontrol, kalite güvence, mühendislik ve bazı durumlarda Ar-Ge bölümleri bir masa etrafında toplanmalı ve en kötü koşulları belirleyerek bu testleri, bu deneyleri kurgulamalıdır diyor. Test edilen materyalin testlerde spesifikasyon limitlerinde kullanılması, bitmiş ürün gerekli spesifikasyonlarda üretmek için materyallerin prosesi sürdürebileceğinin bir işareti olabilir diye de bunu izah ediyor. Yine ISO bakın çok kapsamlı olarak bir filtre seçimine karar verme aşamasında özellikle filtrasyonla ilgili bölümde o kadar çok etki eden parametre var ki bunları tek tek sayıyor. Biz bunları şu şekilde alt gruplara ayırdık sadece. O alttaki çıkan fiziko-kimyasal, biyolojik yük, filtrasyon parametreleri ve ortam koşullarını biz ekledik sadece. Ama diğer parametreler tamamen ISO standartının içerisinden alınmış maddelerdir. pH, viskozite, iyonik güç, ozmolarite. Yani bir filtre seçeceğim. Seçerken hangi kriterleri göz önünde bulundurmam lazım? En kötü koşulları belirlerken bunlara dikkat etmem gerekiyor. Yüzey aktivitesi gerilimi, sıvının mikroorganizmalardan etkisi, filtrasyon süresi, kullanılan filtre birim alanı başına filtrasyon hacmi, akış hızı, fark basıncı ki bunlarla ilgili teknik olarak Ali arkadaşım birazdan daha detaylı bilgiler aktaracak. Madde madde baktığımızda sıcaklık, sterilizasyon koşulları, tekrarlanan sterilizasyonların etkisinin uygunluğu gibi birçok faktör göz önünde bulundurulmalıdır. Yine EMA özellikle ruhsat başvurularında sağlanacak bilgi ve veriler kısmında siz laboratuvar ölçeğinde çeşitli deneylerle yaptığınız, seçtiğiniz filtreyi proses tarafında da scale-up'a gittiğinizde uygulanabilir olmasını sizden istiyor. Burada aslında şöyle bir izahatta bulunabilirim. Şimdi laboratuvar ölçeğinde daha mini boyutlarda çeşitli filtre materyalleriyle bazı denemeler yapılıyor her ilaç firmasında. Dolayısıyla bu seçimi yaparken şimdi laboratuvarda kullandığınız bir filtreyi düşünecek olursak bunun proseste de aynı filtreden olması gerekiyor. Bunu yine bir örnekle şöyle kısaca izah edeyim. Örneğin selüloz asetat veya selüloz nitrat, selüloz nitrat filtre materyali laboratuvar ölçeğinde mevcut. Ama proses boyutunda yok. Dolayısıyla rejenere selüloz veya aynı şekilde benzer şekilde ki çok sıklıkla kullanılıyor. Evet. Rejenere selüloz şirin ucu filtreyle işte biz bir filtre çalışması yaptık. Sonra proses tarafında filtreyi belirlerken aynı filtre materyalin ile orada proses boyutuna geçmemiz lazım. Ama maalesef o tip boyutta, proses boyutunda bir rejenere selüloz materyalde filtre mevcut değil. Bunun gibi scale-up çalışmalarında da bunu birebir uygulanabilir olması gerekli olduğunu söylüyor. Yine çok önemli FDA denetleme kılavuzunda özellikle dozaj form üreticilerin denetlenmesine dair kılavuz bu bakın. Yani denetçileri diyor ki steril filtrasyon kullanıldığı durumlarda yani bir firmaya gittiniz, bir denetleme yapıyorsunuz. Burada bir steril filtrasyon kullanıldığını biliyoruz. Bu firmanın filtre seçimini ve filtre değiştirme sıklığını belirlemek için kullandığı kriterleri sorgulayın diyor. Yani filtre validasyon verilerini bir gözden geçirin diyor. Dolayısıyla bakın burada filtre validasyon verileri diyor. Filtre validasyonu dediğimizde de diğer başta söylediğimiz filtre seçimi ilk başlangıcı, bakteriye yükleme testi, kimyasal uyumluluk testi, extractable, leachable çalışmaları bir bütünsel olarak üçgen piramit göstermiştik. Oradan yine tekrar hafızalara geri getirelim. Dolayısıyla bir denetçi gittiğinde firmanın filtre seçim kriterlerini, filtre validasyon verilerini mutlaka gözden geçirin diye kendi denetçilerine bu konuda FDA bir kılavuzunda bahsediyor. Firmanın ilacın biyolojik yükünü bilip bilmediğini belirleyin diyor. Filtre sağlamlık test prosedürlerini inceleyin diyor. Her üretim sonrasında filtreler değiştirmiyor olabilir. Peki filtrelerin kullanıldığı süre boyunca sağlamlığını koruyup korumadığını, herhangi bir pasajlama olup olmadığını kanıtlayacak veriler olup olmadığını da mutlaka belirleyin diye kendi denetçilerine söylüyor. Ve sonuç olarak filtrasyon prosesinin optimizasyonu yani doğru filtre seçimiyle filtre validasyonu başlamalı. Filtre seçim kriterleri de başta da söylediğimiz gibi çeşitli testler neticesinde otoriteye açıklanabilir olmalıdır. Ben özellikle bu kısımda kendi anlatacak olduğum kısımları bitirdim. Şimdi sözü daha teknik olarak işin başında laboratuvar tarafında olan ve birçok firmanın ürünleriyle ilgili çalışmaları yapmış olan, filtre seçim çalışmalarını yapmış olan arkadaşım Doktor Ali'ye bırakıyorum. Evet Ali buyur.
Çok teşekkür ederim Erol Bey. Buradan sonra ben size biraz daha teknik kısmından bahsedeceğim işin. Piyasada bulunan ürünleri sağ tarafta sizlere listeledik. Sol tarafta listeledik. Sağ tarafta ise filtre üreticileri tarafından üretilen hem final filtre olarak kullanılan hem de ön filtre olarak kullanılan materyalleri görüyorsunuz. Peki bu ürünleri piyasada bulunan ilaç üreticilerinin üretmiş olduğu ürünleri hangi filtrelerle süzebilirim? Yani hangi filtre materyaliyle ben bu ürünleri rahatlıkla sorunsuz bir şekilde kayıpsız, etkinimi kaybetmeden filtreleyebilirim. Bakacak olursak burada pek çok filtre materyali pek çok ürün için kullanılabilmektedir. Veya şunu ben size sorayım. Bir tane filtre materyaliyle birçok ürünü süzebilir miyiz? Böyle bir şey mümkün müdür? Böyle bir şey günümüz teknolojisinde pek mümkün değil. Hatta filtrasyon prosesinin ihtiyaçları, ürünlerin ihtiyaçları, yani ürün içerikleri, ürünle filtre olarak kullanacağınız materyalin polimer özelliklerinin uyumlu olması gerekmektedir. Bundan dolayı filtrasyon teknolojileri sürekli gelişmekte ve yeni polimer materyalleri veya modifiye polimer materyalleri piyasaya sürülmektedir. Bundaki amaç sizin prosesinizdeki ihtiyaçlar ve prosesinizdeki termal dirence, mesela dayanıklı, daha dayanıklı ve etkileşimi az ürünlerin piyasaya sürülmesidir. Bundan dolayı doğru olan şey aslında ürün özellikleri, ürün karakteri, yani filtrelenecek likitin karakteri belirlenmeli, bileşenleri belirlenmeli ve bununla uyumlu polimerler seçilmeli. Bunun yanında seçeceğim polimer prosese göre, yani proses sıcaklığı, proses kontak süresi, prosesteki limitlere, prosesteki pH'a uygun olarak kalifiye edilmeli ve bu şekilde filtre validasyon sürecine ilerlenmelidir. Yine Erol Bey'in de belirttiği gibi regülasyon bu tarz çalışmaları kullanıcılardan beklemektedir. Peki filtre seçiminde, doğru filtre seçiminde kritik parametrelere örnekler verecek olursak bunlar nelerdir? Bir kere filtre materyali, akış hızı, filtrasyon alanı ve filtrenin toplam süzebileceği miktar birbirleriyle doğrudan bağlantılı kritik parametrelerdendir. Likitle, yani filtre edeceğim ürünle seçeceğim filtre materyali birbiriyle uyumlu ise zaten en başlangıçta uygun bir akış performansı yakalanmış olur. Yani daha rahat ve hızlı bir filtrasyonu bu uyumlu materyalle sağlayabiliriz. Bunun yanında uygun akış performansını sağlamamız filtre materyalini de fazla miktarda kullanmamız değil, aslında azaltmamızı, yani küçük boy filtreyle maksimum filtrasyonu sağlamamızı sağlayacağından daha efektif bir filtrasyon elde edilmiş olur. Buradaki kazanım şudur: yüzey alanı arttıkça absorpsiyon oranı, yani etken yardımcı madde kaybım artacağından filtre boyu küçüldükçe aslında bu kayıplar da azalacaktır. Aynı zamanda filtre üzerinde kalan ürün miktarı da azalacaktır. Bu gibi avantajları en başında doğru filtre materyalinin belirlenmesiyle sağlamış oluruz. Bunun yanında doğru filtre materyali seçiminde aslında prosesteki dolum makinesinin veya prosesteki şartların bir arada değerlendirilip filtre boyutunun da buna göre belirlenmesi gerekmektedir. Yani gereksiz yere büyük boy bir filtreyi prosese koyup kontak alanını arttırıp bir risk almak uygun bir sonuç değildir. Regülasyona da zaten açıklanabilecek bir durumda değildir. Devam edecek olursak sıcaklık, viskozite, basınç ve ozmolalite yine birbirleriyle bağlantılı kritik parametrelerdendir. Üreticiler viskozitesi yüksek, yağlı veya emisyon gibi ürünlerde sıcaklık ve basınç artışını uygun bir akış performansı elde etmek için kullanabilir. Bu bizlerin de çalışmalarımızda denediği bir yöntemdir. Fakat sıcaklık ve basınç değişiminde çok dikkatli olunmalıdır. Sıcaklık artışıyla sağ taraftaki grafikte göreceğiniz üzere ürünün yüzey geriliminde ve viskozitesinde bir düşüş sağlanabilir. Ve basıncın da artışıyla hızlı bir filtrasyon sağlanmış olur. Ama PDA Teknik Report 26'nın da üzerinde durduğu gibi sıcaklık artışı absorptif etkiyi tetikleyebilir ve üründeki etken kaybını arttırabilir. Bu konuda dikkatli olunması gerekmektedir ve sıcaklık artışıyla bir ürün stabilitesinde de çeşitli problemlerle karşılaşılabilir. Bu da yine optimize edilmesi gereken bir noktadır. Yine viskozitesi yüksek ürünlerde ve ozmolalitesi yüksek ürünlerde bakteri morfolojisi olağan formundan daha küçük bir forma kavuşacağından dolayı basınç artışıyla bir küçük olan bakteri, küçük olan yine porlara, filtre üzerindeki gözeneklere zorlanmış olur ve bu tarz proseslerde sterilite de bir soru işaretidir. Bu gibi proseslerde filtrasyon yüzey alanı arttırılıp basınç değerleri aşağıya çekilerek aslında riskler aşağıya çekilmiş veya elimine edilmiş olabilir. Bu konuda yine değerlendirilip bu kriterlerin dokümante edilmesi gerekmektedir. pH, iyonik yük, absorpsiyon birbirleriyle bağlantılı parametrelerdendir. Yine PDA Teknik Report 26'da sıcaklığın yanında PDA Teknik Report'un absorpsiyon başlığı altında belirttiği noktalardan bir tanesi de pH'tır. pH değişikliği iyonik gücü değiştireceğinden hiç beklenmedik bir absorpsiyona sebep olabilir. Yani ürün ve polimer yapısı birbirine tutunup etkeninizi veya yardımcı maddelerinizi üründen uzaklaştırmış, filtre üzerinde bırakmış ve final ürününüzde bir kalite kaybıyla karşılaşmış olabilirsiniz. Bunun yanında pH'in şöyle bir efekti de vardır. pH değişikliği yapıldığı takdirde eğer ürünün pH aralığı filtrenin çalışma pH aralığına uygun değilse filtre iki tipte tutuş sağlar. Yani eleme sağlar üründen. Bir, statik olarak, fiziksel olarak tutmak. İki, iyonik güç ile tutmak. İyonik güçle tutma özelliğini pH değişikliğinden dolayı ya da pH'ın uygun aralıkta olmamasından dolayı filtrenin çalışma pH'sına bakteri tutma özelliğini filtrenin kaybedebiliriz bu tip ürünlerde ve filtre aslında sterilizasyonu sağlamaz. Üründe biyolojik bir yük varsa filtreden sonraki final ürüne de geçer ve burada sterilite bir soru işaretidir. Yani boş yere bir filtre kullanmış oluruz. Bu sebepten dolayı doğru filtre seçimi en başta yapılmalıdır ve Erol Bey'in de belirttiği gibi daha sonra seçilen doğru filtre kalifikasyon dokümanları da varlığında desteklenmelidir. Yani filtremin çalışma pH'sı, filtremin prosese uygun sıcaklıkta çalışıp çalışmadığının dokümantasyonu yapılmalıdır. Az önce saydığım kritik parametreleri yine doğru filtre seçiminde dikkat edilmesi gereken parametreler başlığı altında sizlere sadeleştirerek en önemlilerini topladık. Burada doğru filtre seçiminde 35 temel ana parametreyi sizlere sunduk ve bunları da 7 ana başlık altında topladık. Bunların her biri çok kritik, çok önemli parametrelerken bu parametrelerin birbirleri varlığındaki davranışları farklılık göstermektedir. Örneğin viskozitesi yüksek ürünlerde sıcaklık ve basınç artışı uygulandığında proseste sıcaklık artışıyla sterilite ve leachable'da sıcaklık artışıyla leachable'da bir sıkıntı yaşayabiliriz. Yani leachable nedir? Ürünümden herhangi bir şey, filtremden herhangi bir şey ürünüme bulaşabilir. Yani polimer yapısından bir şeyi sıcaklık artışıyla bir ürünüm çözebilir ve final ürünümde istemediğim bir polimer materyali görebiliriz ve bu da final ürünümün kalitesini etkileyen bir durumdur. Bunun yanında yine basınç artışıyla bir viskoz ürünlerde bakteri morfolojisi küçüleceğinden dolayı sterilitede bir soru işareti olabilir. Yani final ürününde steriliteyi sağlayamayabilirim. Bunun yanında yine sıcaklık parametresi ve pH parametresi bu parametrelere göre bizim ürünümüzdeki etken ve yardımcı madde filtre üzerinde tutulabilir. Eğer uygun proses değerlerini sağlayamazsam. Bu tamamen ürün varlığında ve filtre varlığında yapılan simülasyon çalışmalarıyla, denemelerle analiz edilecek ve dokümante edilecek bir çalışmadır. Bu da yine önemli kritik parametrelerdendir. Bunun yanında yine kimyasal uyumluluğu, seçtiğim filtre materyalinin filtre edeceğim ürünle kimyasal uyumluluğu. Bu en önemli noktalardan bir tanesidir. Bu da sağlanmadığı takdirde etken yardımcı madde kaybı yaşayabilirim. Aynı zamanda yanlış materyal seçtiğimde akış hızı da düşebilir. Akış hızı düşmesiyle bir daha fazla filtrasyon alanı kullanabilirim. Ve bununla birlikte uyumluluk, kimyasal uyumluluk olmadığından dolayı final ürünümde partikül de görebilirim. Yani yanlış seçimlerin aslında nelere sebep olduğunu, nelerin neleri tetiklediğini aslında bir arada burada görüyoruz. Dikkat edecek olursak buradaki her parametre birbiriyle yakinen bağlantılı parametrelerdir. Aslında bir seçim programı izlenmeli ve regülasyonu da belirttiği üzere prosesteki ihtiyaçlar, ürünün ihtiyaçları bir arada değerlendirilip çeşitli laboratuvar ölçekli simülasyon çalışmalarıyla biz doğru filtre seçimini, regülasyonu açıklamalıyız. Çoğu kez karşılaştığımız bir durumdur. Biz filtre tedarikçilerine genelde sorulur. Şu tipte bir ürünüm var ve bu ürünüm şu bileşenlerden oluşur. Bu ürün için hangi filtre materyalini kullanabilirim? Keza Erol Bey'e de çok sorulmuştur. Bizlere de çok yöneltilmiştir bu tarz sorular. Burada aslında doğru olan şey kişilerin filtre materyalini belirlemesi değil, ürünün özelliklerini ve prosesteki ihtiyaçların bir arada değerlendirilerek doğru filtre materyalinin seçimidir. Regülasyon bunu beklemektedir.
Biz de burada bir takip programı çerçevesinde bu işi yapıyoruz.
Evet.
Onunla ilgili de izlediğimiz yol şu anda.
Aynen. Burada aslında proseste dikkat edilmesi gerekenler. Öncelikle biz ürünün özelliklerini, yani ürünün sahip olduğu etken, koruyucusu, solventi, yardımcı maddeleri bir arada değerlendirilerek filtre adayları seçilir aslında ilk etapta. Sonra ürün karakteri, ürün özellikleri, ürünün fiziko-kimyasal özellikleri, prosesteki ihtiyaçlar, proses limitleri, üretim hacmi, proses sıcaklığı, giriş basıncı, kontak süresi ya da dolum makinesi performansı, hangi debiye ihtiyacım var benim proseste? Bu tarz bilgiler bir arada değerlendirilerek aslında bir filtre seçim programı oluşturulur. Biz yaptığımız çalışmalarda Sartorius patentli Zero T ekipmanını kullanıyoruz. Bu çalışmalar şu şekilde oluşmakta. Bu ekipman da iki kısımdan oluşuyor. Bir taraf yazılım kısmı, elde ettiğimiz verileri analiz etmemizi sağlayan ve teknik ham dataları alıp teknik olarak analiz etmemizi sağlayan kısımdır. Diğer kısım ise prosesteki veriler dahilinde simülasyon çalışması yaptığımız, yani sahip olduğunuz paslanmaz çelik ekipman, single use ekipman, basınçla ittirme mi yoksa peristaltik pompayla veya pompayla mı süzüm yapılıyor? Gamma steril ürün mü kullanılıyor yoksa çoklu kullanıma sahip prosesteki bileşenlere mi sahipsiniz? Kullanılan silikon hortumlar, teflon hortumlar vesaire tüm proses komponentleri birlikte simüle edilerek çalışmalar ele alınıyor. Bu simülasyon çalışmasında birbirinden farklı filtre ve filtre kombinasyonları denenerek filtrasyon akış trendleri, toplam süzüm trendleri ve basınç trendleri bir arada değerlendiriliyor ve bunlara ait ham datalar elde ediliyor. Bununla kalmayıp filtrasyon sonrasında filtrenin sağlam olup olmadığı daha sonra yine bizler tarafından integrit test yöntemiyle Sartoçek cihazlarıyla test ediliyor ve filtre hala mukavemetini korumuyorsa zaten o filtre o testten kalmış oluyor. Yapılan bu simülasyon çalışmalarında sadece fiziksel testler yapılmıyor, aynı zamanda analitik testler de yapılıyor. Yani filtrasyon öncesinde ve filtrasyon sonrasında bu proses simülasyonu denemelerinde çeşitli numuneler alınıyor. Ürünün kendisinden örnekler alınıyor. Filtrasyon öncesindeki numuneye, yani kontrol numuneme göre filtrasyon sonrasındaki çeşitli filtrelerden filtreleyip aldığım numunelerimde etken kaybı var mı, fiziko-kimyasal değişiklikler var mı veya herhangi bir impürite veya bir safsızlık, partikül görülmesi gibi bir durum var mı? Bunlar analiz ediliyor ve filtre seçimleri bu şekilde değerlendiriliyor. Bu filtre değerlendirme programında şu da çok önemli bir durumdur. Birden fazla filtre ve filtre kombinasyonları denenmekte. Yani ön filtre artı final filtre kombinasyonları denenmekte. Biz denemelerimizde ve piyasadaki hakim olduğumuz konularda şunu görüyoruz. Bazı ürünlerin filtrasyonunda hiç gerekmediği halde fazla filtre materyali kullanılıyor. Büyük boy filtre kullanılıyor, gereksiz yere ön filtre kullanılıyor. Bu tarz simülasyon çalışmaları bu filtre materyallerinin ne kadarının gerekli olup ne kadarının gerekli olmadığını ve prosesinizdeki fazla kontak alanlarını azaltmanızı, validasyona daha emin adımlarla gitmenizi sağlayacak kritik çalışmalardandır.
Bazen Ali şöyle diyeyim, çok
Tabii,
şöyle düşünülebiliyor. Yani filtre biraz büyük kullanalım, daha süzüm olsun şeklinde. Ama bunu tabii diğer taraflara yansıması var, öyle değil mi?
Bu kesinlikle öyle Erol Bey. Yani yıllardır üretim yapan üretimciler dahi büyük filtre olsun ben rahat filtrasyon yapayım diyorlar. Size de denk gelmiştir. Yani bu aslında doğru bir şey değildir. Validasyon çalışmalarında bu filtrenin başarısız olma ihtimali vardır. Doğru filtre materyali olsa bile. Çünkü proseste absorpsiyon görebiliriz. Etken kaybı var. Excess doz kullanılıyor ki regülasyon artık bunu da çok ciddi olarak irdeliyor ve neden kullanıyorsun? Final ürününde bu excess kalıyor mu? Bunları hep sorgulamakta. Bu tamamen yanlış boyutlandırmadan kaynaklı olan şeyler aslında. Bunları da aslında burada bu bilgileri paylaşmamız çoğu kişinin prosesindeki hataları analiz etmesi için de önemli. Özellikle bunları söylemek istiyorum. Biz yaptığımız bu çalışmalarda aynı zamanda yazılımsal olarak doğru filtre boyutunu yine proses limitleri dahilinde belirliyoruz. Yine akış trendlerine göre bu neyi sağlıyor? Doğru filtre materyalini seçseniz fakat yanlış filtre boyutunu kullansanız dahi prosesinizde risk vardır. Az önce söylediğim nedenlerden ötürü. Büyük boy filtre fazla kontak alanı demektir. Bu da sizin prosesinizde leachable olmasını, etken kaybını, hatta final filtrasyon yapıldıktan sonra filtre üzerinde kalan ürün kaybını doğrudan etkileyen kritik noktalardandır. Doğru filtre seçimi regülasyonlara göre nasıl yapılmalıdır? Regülasyonun beklentisi nedir? Kalifikasyon verileri, prosesteki ihtiyaçlar ve ürün özellikleri bir arada değerlendirilerek öncelikle aday filtreler belirlenmelidir. Daha sonra belirlenen aday filtrelerle bir takım simülasyon çalışmaları laboratuvar ölçeğinde yapılmalı ve proses efektifliği sağlanmalı. Buradaki denemelerde gerekiyorsa ön filtre basamağı yani ekstradan bir koruyucu filtre basamağı prosese eklenebilir. Bununla birlikte aday filtreler fiziko-kimyasal olarak, analitik olarak değerlendirmeli ve bunlardan uygun olanlar filtrability performansı bakımından iyi olanlar seçilmeli. Bu iyi olan filtre materyali ile birlikte optimizasyon çalışmaları yapılmalı ve finalde de bu regülasyonun beklentisi üzerine dokümante edilmeli ve raporlanmalıdır. Yani çalışmayı yapıyorum fakat bu çalışmayı da dokümante edip regülasyona açıklamam gerekmekte. Peki ürünü özel doğru filtre seçiminde neleri takip etmeliyim? Bunları tabii ilaç üreticileri risk analiziyle yapmalıdır. Prosesteki riskler neler olabilir? Bunları inceleyecek olursak filtre seçiminde etken madde analizi yapmalı mıyım? Bunu zaten Erol Bey de açıkladı. Ben de tekrardan açıklayayım. Regülasyon söylüyor ki filtre edilen ürün filtreyi etkilememeli. Aynı zamanda filtreden de etkilenmemeli. Yani yapısından herhangi bir şey filtreye de bırakmamalı. Aynı zamanda filtreden de bir şey çözüp içine almamalı. Bu filtreden beklenendir. Bu kapsamda da filtrasyon öncesinde ve filtrasyon sonrasında yapılacak absorpsiyon testleriyle, fiziko-kimyasal testlerle, analitik testlerle bu konu regülasyona açıklanmalıdır. Peki yanlış filtreyi seçersem proseste ne tür aksaklıklarla karşılaşabilirim? Yanlış filtre materyali seçildiğinde bu olabilecek en kötü senaryolardan bir tanesidir. Filtre validasyonu süreci yanlış filtreyle tamamlanamaz. Leachable testinden veya chemical compatibility testinden, kimyasal uyumluluk testinden, bakteriye challenge testten ilerleyen süreçte bu filtre kalabilir ve bu şekilde ruhsat dokümanı ile yine ruhsat başvurusuna başvurulmaz, ruhsatta kabul edilemez. Risk analizimi hangi ana parametre üzerinde durarak yapmalıyım? Regülasyonun beklentisi aslında bir parametre değil, proseste yer alan parametreler ve risk analizi ile bu parametrelerin değerlendirilmesi ve buna göre filtrenin seçilmesi ve seçilen filtre ile prosesteki bütünlüğün sağlanması, yani proses sonrasında herhangi bir şey final ürününde görüp görmediğimiz bu proses limitlerinde regülasyona kanıtlanmalıdır. Seçeceğim filtre ne kadar ürün kaybına yol açacak? Seçtiğimiz filtre materyalinin önemi kadar aslında doğru boyutlandırılması da önemlidir. Çünkü yanlış boyutlandırma yaptığımızda bir boy büyük filtre materyalini belirlediğimizde proses başına minimum 200 mililitre hatta filtre üzerinde kalabilir. 200 mililitre az bir miktar ürün gibi görünse bile ürünlerin bazılarının mililitresi bile çok kıymetli olabiliyor. Bu tarz kıymetli ürünlerde 200 mililitre validasyon süreci giderlerini bile çok rahatlıkla karşılayabilir. Hatta prosesteki ardışık olarak üretimden kaynaklı kayıplar aslında üretim maliyetlerinin de çok üstüne çıkmaktadır bu tarz şeylerde. Bundan dolayı doğru filtre materyali seçimi kadar doğru boyutlandırma da önemlidir ve kritiktir. Denetimde filtre seçimini denetçiye nasıl açıklayacağım? Erol Bey zaten bu konuda özetledi. Denetçilerden beklenenler, yani denetçilerin soracağı soruları size bizzat göstermiş oldu ve sunmuş oldu. Denetçiler bugün piyasada görüyoruz ki filtre kaynaklı bir absorpsiyon, filtrenin sterilizasyonu sağladığına dair, yani filtrenin integrity testindeki değerlerin dahi bakteri tutma kapasitesiyle korele olup olmadığının açıklanması, dokümante edilmesi, filtre kaynaklı gelecek maddeler, extractable dataları vesaire bilgileri kullanıcılardan beklemektedir. Bu tarz dokümantasyonların yapılmasını takiben dokümantasyonlarla regülasyona açıklamalar yapılmalıdır. Ben sunumumun sonuna yaklaşırken size kritik bir tane örnek vermek istiyorum. Bizim karşılaştığımız bir konu, yanlış proseste yanlış filtre seçimine bir örnek. Proseste yanlış filtre materyali seçildiğinde ne olabilir? Büyük üreticilerden bir tanesinin 600 litrelik bir üretim var. Bu arada şunu açıklayayım, bu bir senaryo değildir. Tamamen yaşanmış bir olaydır. Gerçek büyük üretici bir firmanın bizzat prosesinde yaşadığı bir sorundur. Bu 600 litrelik bir üretimi var bu üreticinin. Üretimi 3 tane 10 inç ön filtre ve 3 tane 10 inç final filtre ile yapması bekleniyor normalde. Prosesi bu şekilde optimize etmişler. Fakat bu üretici üretime giriyor ve 600 litreye boyutlandırıyor ve 600 litrelik üretimini yaparken prosesinde bir tıkanmayla karşılaşıyor ve yeni bir set filtre ile değiştiriyor bu filtreleri. Yine 600 litrelik üretim yeni setle bile yapılamıyor, tamamlanamıyor ve 3. setle proseste ısıtma da likite, yani ilaca ısıtma işlemi de uygulayarak zorlukla tamamlayabiliyorlar bu prosesi. Daha sonra üretici bu yaşadığı problemden dolayı ürünü alıyor ve bize getiriyor. Bu tipte bir ürünüm var, ben proseste tıkanma yaşıyorum ve filtre validasyonunu da yaptırmak istiyorum. Bize yardımcı olabilir misiniz diye. Biz aldığımız ürünle laboratuvar çalışmaları yapıyoruz ve denemelerimiz dahilinde aslında yanlış filtre materyalinin seçildiği, ön filtre basamağına dahi gerek olmadığını, bir tane 10 inç filtre ile 600 litrelik filtrasyon yapılabileceğini belirliyoruz ve buradaki tabloyu siz net olarak görüyorsunuz. Yani burada prosesteki kayıplar, fazla sarf kullanımı, ondan sonra operasyonel maliyetler, pek çok şey var. Aslında ben bu kayıpların detayına da çok girmek istemiyorum. Aslında tablo zaten, evet, tamamen her şeyi gösteriyor. Burada aslında kullanıcının tamamen seçimleri, alacağı doğru kararlar kendi prosesine yön vereceğinin bir göstergesi bu.
Bu tür maalesef hatalarla veya bu tür yanlışlarla hani böyle sadece bir firmada da değil, zaman zaman karşılaşabiliyoruz.
Yani yüzlerce karşılaştığımız.
Dolayısıyla aslında filtre seçimini bugün biz anlatmaya çalıştık Ali arkadaşımla birlikte. Şöyle genel bir yine toparlayacak olursak, sorular da varsa bu arada soruları da alabiliriz. Çok fazla bir vaktimiz kalmadı. Ek 5 dakikalık bir süremiz var. Ancak filtre seçiminin özetle söyleyecek olursak, eskiden kalan alışkanlıkla işte bizim şöyle bir ürünümüz var, bu jenerik bir ürün. A firmasında da var, B firmasında da var. Dolayısıyla orada ne kullanılıyorsa biz de onu kullanalım şeklinde bu tür geçmişte birçok çalışan arkadaşlarımıza sorular gelmiştir. Biz de tabii ki yardımcı olmaya çalışmışızdır. Ancak özellikle bu seçimin karar verme noktasında regülasyonlara göre, otoritelerin isteğine göre nasıl yapılması gerekli olduğu, özellikle yine tekrar ediyorum 2018 1 Ocak itibariyle PIX üyeliğiyle biraz daha önem kazandı ve bir zorunluluk haline geldi. Diğer taraftan biz bunu zaten şöyle söyleyeyim 15 yıldır zaten yurt dışına ürün üreten üreticilere zaman zaman bazı ürünlerinde bu tür çalışmaları zaten yapıyor idik. Ancak PIX üyeliğiyle bu artık bütün firmalar için zorunluluk haline gelmiştir. Dolayısıyla bu seçim kriterleri otoriteye çok iyi izah edilmelidir. Bir filtre seçim programı takip edilerek en kötü koşullarda testler, deneyler kurgulayarak bu özellikle istenen kriterleri sağlayan filtreyi bu çerçevede seçip ona göre dokümante edilmelidir. Bu şekilde yapılmalıdır. Aksi durumda gerek bir üretim esnasında işte yaşanmış sıkıntıyı örnek olarak size ilettik son kısımda Ali arkadaşım ki bunları dediğim gibi sayısı onlarcadır maalesef. Dolayısıyla hem maddi kayıplar hem prosesteki çok ciddi zaman kayıpları, diğer bir sürü altına madde ekleyebilirsiniz. Çok ciddi sıkıntılara yol açabilmektedir ki kaldı ki özellikle ilaç camiasındaki üreticiler, kullanıcılar bunu çok yakından bilir. Bir aseptik filtrasyonda yaşanacak bir sıkıntı en basitinden değiştirilecek bir filtre bile yani seçtiğimiz bir filtre ile bir dolum süresini bitirmek isteriz. Orada bir filtre değişikliği bile yapılmak istense çok ciddi sıkıntı, zahmet, problem, çeşitli kontaminasyon risklerini barındırmaktadır. Dolayısıyla takip edilen filtre seçiminde takip edilmesi gereken yol gerek otorite nezdinde bu şekilde izah edebilmekteyiz.
Bizler sizlere teşekkür ediyoruz katılımlarınızdan dolayı. Sorularımız varsa alabiliriz. Herhangi bir soru yok anladığımız kadarıyla. Yani bu süre zarfı içerisinde biz yani elimizden geldiğince bu konuları anlattık. Şu an için.
Evet, bu sunum tabii ki kısıtlı bir zaman şeyimiz var. Biz burada sadece başta da söylediğimiz gibi filtre validasyonu kapsamında en başlangıçta yapılması gerekli olan ürüne ve proses parametrelerine göre uygun filtre tipinin, ebadının, materyalinin belirlenmesinde izlenmesi gereken yolları kısaca anlatmaya çalıştık. Ama daha detaylı bu filtre validasyonu ile ilgili bilgi talepleriniz olması halinde bize sartonet@sartonet.com'dan ulaşabilirsiniz. Dolayısıyla hani firma bazında veya kişisel bazda sorularınız olduğunda da her zaman destek olacağımızı, yardımcı olacağımızı bildirmek isteriz. Hepinize sunuma katıldığınız için teşekkür ederiz. İyi fuarlar, BioExpo çerçevesinde iyi fuarlar diliyoruz. İyi sunumlar diliyoruz. Hepinize iyi günler diliyoruz.
İyi günler. Teşekkür ederiz.